So today we have two talks that will be at the systems level. And we saw talks yesterday as well that there's a lot of progress being made in both analyzing data, conceptual frameworks, and so we hope to get a good discussion going. Barry is at NIH, where he has a monkey lab that has been pioneering in studying divisional system, and also parts of the memory system that's farther up the hierarchy, and also of all things, addiction.
Oh, reward.
Okay, reward. Addicting rewards. Okay. In any case, he said he never got the monkeys to work nearly as hard as they did with instead of a drop of juice, they were giving a drop of opioid, right?
Okay.
Cocaine.
Bobby, what do you think tonight? Can we try that?
Okay, who's going to supply it?
I never did that experiment. It was just too much trouble to handle restricted drugs.
Oh, okay. Well, even the NIH has problems with restrictions. But in any case, you're going to hear today about the representation of complex images in the brain, the ventral visual stream.
Okay, and maybe I'll get to doing a little more of it. The NIH in their magnificence has locked me out of Zoom sometime in a day or two. I had to run down to my condo to get my government ID.
Do you Zoom?
To use my computer. Well, to use Zoom, to log into things. They're very restrictive about things. Anyway, we're up and going. For people who were here last year, the first about quarter to third of this talk is going to be similar, if not identical, to what I talked about last year. But to explain what we've done in the past year, I need to make sure you learn something about what we were doing in general and where we were a year ago. I hope this starts a big discussion with Uncle Barry and Gerald Powell. I think it'll be interesting to see if some of the things we're talking about can be cast in wider terms. Okay, so the first thing I'm going to do is give you a hope. All right, wake up.
So is this monitor supposed to be on? Is it?
It needs to be set, so you'll do that after the talk.
Okay, so the first thing is I'm going to give everyone a little mini version of the visual system in the monkey, which we think is a fair representation of the visual system in humans. So one of the great things that, whether you know you're assuming this or not, you're almost surely assuming that there's functional localization. And one of the ways I can demonstrate is we talk about visual cortex, we talk about auditory cortex, we talk about somatosensory cortex. Those are primary sensory cortices, but it illustrates the point that we think there's some restriction and speculation of function as you move around this big mass of tissue that we call the brain. A great deal of what we know about functional localization, the fact that visual cortex is in some sense visual and not auditory. And I don't say that in any absolute terms. And it comes from brain injuries. People observed that if you get a local lesion in your brain, say in primary visual cortex, you have a great deal of trouble seeing things in a localized part of space. And we think of that as visual fields, and mapping of visual fields. Just as an aside, One of the big breakthroughs in mapping functional vision in the human came from the development of high velocity jacketed bullets. And that happened in the American Civil War and the Franco-Prussian War of 1870. There was a big change in the kinds of wounds that people got from being struck by a bullet, because now the bullets were jacketed, which meant they didn't fall apart very easily, and they were they moved at much higher velocity, which meant that they made a much more discrete lesion because they went through the brain instead of stopping in the middle, giving up all the energy that they have, all the kinetic energy, and turning the brain into a bowl of jello. And so a lot of what we learned.
So you're saying that there was a great advance for neuroscience.
Yeah.
Well, a side benefit.
Yes, I think it's just an interesting tidbit. This idea of localization or differentiation of the brain into parcels comes from actually looking at tissue. This is a stained section of a monkey brain through the area we call V1, primary visual cortex, at the place where it transitions into V2 or visual area 2. And then they just get numbered. Someone watching on Zoom, do we know? They're not going to be able to see this. Okay. Anyway, here at the
We're the military grade pointer.
I'm getting a picture.
there's this break between primary visual cortex and V2. And what happens is that there's a line of cell nuclei that disappear. You can actually see these on a brain cutting. If you don't ever go in a hospital where the neuropathologist is doing a brain cutting, ask them to take a slice through V1. You can see this stripe called the Stryker genome. It's reasonably.
Now this is stained, right?
Yeah, this is stained, but you can see it on a fresh. The reason I'm going through all this is I'm going to rely heavily on what this diagram embodies, which is, here is this primary visual cortex. Here is what I was calling V2. It's a different terminology. It's an older terminology. It comes from a book by two neuroanatomists, Bonin and Bailey, about 1948. And this is the monkey brain parceled out as they thought it looked when they looked through a microscope. And the claim would be, and notice I couch that cautiously, the claim would be that, for example, you could tell that OA, which is what we know as V4 and TEO, which I'll get to in a few minutes, are here and TE is here. And that if you look at the architecture of the same section, it's going to look different in those two areas. Now, that reason I'm cautious about that is some of that is in the eyes of the beholder. There's no question that trained neuroanatomists are a little more finely tuned for doing this, but some of these lines take their placement with a grain of salt. There's probably no question that if you're at let me orient you. This is the lateral view of a monkey brain. This is the front of the brain. This is the back of the brain. And information comes in through the eye, goes in deep into the lateral geniculate nucleus, and then comes to primary visual cortex, which is at the back. And what I'm going to be talking about is this diagram. All this folderol I just went through. is so I can explain this diagram. So we're interested in higher visual function. And we know that if you make lesions in this, sorry, this got turned around also. So this is now the front and this is the back. If you make lesions in this area that's labeled TE or TEO plus TE, you'll get monkeys that in many aspects have what looks like normal vision, but they have trouble distinguishing one complex object from another. And in fact, in this area TE, there's a great sub-industry in our field right now to study sub-areas within this that are have cells that are sensitive to particular classes of images. The class being of most interest is faces. So in this area are 5 little patches that people argue are rich in face cells. That does not mean that all cells there are most sensitive to faces, but it does mean that they expect if you record there with a microelectrode and show faces in front of the animal while you're recording, you'll get very nice responses. And if you use non-face stimuli, you get less strong responses. So there is an idea that there's a hierarchy in vision that starts in the primary visual cortex, and the cells in the primary visual cortex are sensitive to simple objects, generally stated to be bars and edges. And if you're recording them, Mike Stryker records a lot in this area, maybe almost exclusively.
A lot.
A lot is a fair statement, but it's fun. I mean, when you're doing it, because you get a lot of positive reinforcement from the film, even if you're not at the moment doing an experiment that yields anything new.
Barry, what's 4-H?
Oh, sorry. These areas are this primary visual cortex. This is V4, TEO, which I'm going to talk about. We'll talk TE, and RH is rhinal cortex. And it shows up just at the tip of the frontal lobe. If I finish quickly, which I'm probably not going to do, I was going to show you a little bit on reward processing, because I will make the bold statement that when you're recording in TE or looking for function in TE, I'm going to show you a lot of functional results. If you go to rhinal cortex, you got no deficit in function in object vision, at least in our hands. This is controversial, and people say we don't know what we're doing. But if I do their experiment, we get our result, not theirs. And what happens when you lesion rhinal cortex bilaterally is the animals have trouble distinguishing one reward from another. And if I don't get to it, you want to see it, show it to you.
So it's part of a bigger system, though.
Yeah. So in fact, the way we think of it is this is the visual system. Where does visual information get assigned valence so that it means something emotional to you? You basically never look at anything that doesn't bring up some valence. It may not be very strong, it may not be very cogent, but the idea that you see something, ever see something that has absolutely no meaning to you is probably not the case, or mostly not the case. Anyway, so I'm going to talk about these areas going down the temporal lobe, which have been called a system for high-level vision. And here's two pieces of information. This is, if you think of this as a bucket brigade going from simple objects to complex objects as you go through different parts of the visual system. As you go from back to front, you're going from simpler objects to more complicated objects. And the other thing that happens is the receptive field. That is the area, the part of the visual field that can be stimulated and give a response when you're recording with a microelectrode. Those receptive fields get larger as you move more toward the front of the brain. And the ones in TE, this area at the back or at the very front, that's probably the last purely visual processing area, are bilateral. And I'm going to show you some anatomy in a minute that we've done that shows that it's a bigger deal than we even thought.
So inferotemporal cortex.
Yeah, I don't like that term.
Okay, but
Because people who use that term are usually talking about TE and TEO,
right,
without distinguishing between them. I'm going to show you that's a mistake. At least it seems to be a mistake. So we need a task where the animals have to tell us what they're looking at. And we chose to start with a categorization task, a task where the monkeys are shown a large set of dogs and cats. And they have to tell us, is it a dog or a cat? And when we did this initially, well, let me tell you how, because I put the slides in this order. Let me tell you how we get the monkey to indicate the perceptual judgment. So we teach the monkey a simple task. My joke about this is it's the only task I know how to teach a monkey. And what it is a monkey's looking at a screen, a little spot comes on. At some random time later, it turns green. If the monkey lets go of the bar it's holding, at the time this turns green, the monkey gets a drop of juice. Very simple. And in fact, this task was invented by Bob Wertz to be able to map visual receptive fields in the awake monkey, which he did in the late 60s with this task, and this was the paper.
And how many trials does it take for them to get that?
To do this part of the task takes about 2 weeks, an hour and a half session a day. And you can speed it up if you do what's called hand shaping. If you sit there and really watch carefully what the monkey's doing, you can do it a little quicker. But I'm going to show you something. I'll show you what happens when we do the overlay that we do on this, which is why we do it this way. So what we added to this is we just put up a picture of a cat or a dog at the beginning when the animal touches the bar. The monkey is only trained to do this red-green discrimination. If there's a dog there, the monkey has to hold the bar until the green light comes on, release it and gets a drop of juice. If a cat comes on, the monkey needs to let go of the bar before the spot changes color. So now this would be in classic signal detection theory, a two alternative force choice in time. That's what we teach the animals. And this part of it, you can teach a good monkey in half a session, and a dumb monkey in a couple sessions, and an even dumber monkey visits the anatomist. Yes.
So, do they forget this task? How long does it take them to forget this specific task like this?
The task?
Yeah.
As far as we can tell, they never forget the test once they've learned it. I used to joke, it's not so true right now, But I used to joke I had monkeys that had been in my group longer than the postdocs. Because we were doing a lot of behavioral experiments, and we just used the same monkeys over and over again. And some of them were around for 7 or 8 years.
You could have gotten tenure.
I thought of it, but in fact, some of them were better than some of the people who did it.
But it was always the same picture, right?
No. Let me show you the next slide.
Oh, okay. Right.
This is the training set. This is 20 dogs and 20 cats. And so when we did this, Toby, we didn't do 1 cat and 1 dog. We just threw them in and gave them the 20 dogs and 20 cats. And that's what they learn in half a day.
Have you ever seen these animals in real life? These monkeys, have they ever seen them real dogs and cats?
I can't, probably not, but I can't guarantee that.
Well, where do the monkeys come from?
Our monkeys come from a colony that our lab owns, which you guys don't appreciate what I'm saying, probably. But right now, it's almost impossible to buy a monkey, a rhesus monkey, anywhere in the world.
What? Really?
Yeah, COVID took care of all the available rhesus monkeys. Drug companies bought them all. And 2 large sets we don't have a large group. We have about 40 monkeys. That's a breeding colony. So that they produce the number of monkeys that 5 PIs need on a yearly basis.
So, I mean, monkeys aside, how the heck do you dogs tell that the other thing is a dog? I mean, a Chihuahua from a Great Dane's a dog.
That's why I'm doing it to you guys. You're the guys who are supposed to tell us how you can tell.
No, but they do it. I mean,
Yes, the monkeys do it.
not monkeys, dogs. How
Yeah.
can a Chihuahua tell a Great Dane is a dog?
Well,
That's so exciting.
Look, how does this, my monkeys, how do they know this full face on dog is also a dog like this?
Well, why?
Well, the thing I have to tell you then, once they get this, then they start getting sets of randomly chosen dogs and cats that they've never seen before, and in some experiments will never see again. And they do it. In fact, if you let me get to that, I'll show you they do that. Okay, so we did this dog-cat categorization. The first thing we wanted to do was see whether we could reproduce what was claimed in the literature, but there was no real experiment to back it up, which is monkeys with a rostral lesion of this area of the brain, TE, maybe TEO plus TE. Can't do categorization. Well, it's not quite that simple. Here's comparing the performance of 3 normal monkeys doing this task with trial unique dogs and cats. These are ones they've never seen before, and they never see them again. 240 of them. And as you can see, there's a deficit, but it's not the devastating deficit that we had been led in the literature to expect. Now, maybe that's our fault for overreading the literature, but it was certainly what people thought about this.
Well, maybe it's easier to discriminate some categories than others, right?
Sure. Yeah. And we haven't done anything.
And the fact that they learn so quickly suggests that maybe there might be even some innate circuitry.
Right.
For we know there is for faces, so why not for other animals?
Let's come back to that.
Okay.
Anyway, so we were a little disappointed because we expected the monkeys to be really devastated in this task, and they weren't. So we changed the stimulus set a little bit. And this was the new stimulus set. This is a set of, well, it was a set of, it doesn't have the whole set there, of 20 dog cat pairs that were morphed. So they change gradually from cat to dog as you go along this line. And the monkey's job is still to indicate whether this is a more dog-like image or more cat-like image. And what happens is with the TE lesion, there's a little deficit. Control animals are a little better. And rhinal cortex, this area you asked about, Steve, they're normal. And this is where we're in a little bit of a controversy because I do not think the red and the blue lines are different. Never would be. But there is something else that we saw. The data I'm showing you here, I made a misstatement, which is this is data averaged over 10 days. So they did see that the two sets for 10 days.
How did you reward them?
They get rewarded for indicating the correct category.
But what's the corrective that's in the middle?
Oh, they got a randomly get a reward or not. If you're here.
So if it's a little bit above that, then
they got to take the dog.
A strict boundary between.
Right. The other thing we have done, haven't published, that's fun, is you can move the boundary, say to 75, 25, and they move in a few minutes. So this is reward association, complex image. We haven't made a big deal of that at this point. This was an observation that we're going to make more of in a few minutes, which is, I showed it last year, but what we're seeing is that if you do this 10 days in a row with the same stimulus set, monkeys get better and better. And that's in control animals, animals with TE lesions who are impaired, but they still get better. They are learning something. And the rhinal cortex animals, Steve, I don't know whether you're really interested in them or you just asked the question.
Well, I figured you were going there, so I asked it for the crowd.
Okay, thank you. Anyway, here's the summary of those data. This is the morph level. And this is control animals. This is TEO lesion animals, TE lesion animals. And if you take out both those areas, now you do get a devastating deficit. Those animals really cannot tell one object from another. They look fine if you look at them in the cage, but we were interested in this effect of a partial deficit with TEO lesions or with TE alone. And that when you did them together, did the cumulative lesion, they were really devastated. And so one of the hypotheses was this was a mislabeling of brain areas. That TEO shouldn't be separated from TE. And at this point, we thought TEO and TE had additive effects. Well, that turns out to be, we did another experiment. It's funny how doing another experiment tells you how long you did. And now we changed the task, and this does take more effort to teach the monkeys, but what they're doing is what we would call a recognition task. So they're being shown a series of pictures, and they only see each picture shows exactly twice. The first time when it's unique, and the second time when it's familiar. And the number of unique images between a pair of familiar images in the data I'm going to show you can be anything from 1 to 128. So we'll get some sort of a sense of how well they're doing at remembering. Yeah.
So what is a non-unique image? You have to grant it, you have to find the animal profile.
We just went to the internet and got thousands of pictures.
No,
But it's trial unique, right?
Those are trial unique.
okay, what is a non-unique image?
It's one that had appeared once before
Ah, I see.
in the series,
Just once.
just once.
Okay,
And the question is, do they recognize the difference between never having seen it
I see.
and having seen it once in the past? Okay, everyone with me on that?
And how well do humans do?
Not quite as well as monkeys. You do pretty well. It's surprising.
They need cocaine.
Yeah. Okay, so here's what the data look like. Here's the control data. This is percent correct across all spacings. And they do pretty well at that. Here's the group with the TEO removals. They're unimpaired in this test. Here's TE, they're pretty badly impaired actually. And here's TE, TEO, and they're about the same as TE. Now, let me see what slide.
And 50% is chance?
Yes.
Wait. Percent correct. Is it percent or a fraction? I mean, if it's percent correct, these are very low.
Oh, no, it should be fraction, I think.
Fraction, yeah.
Thank you. And here it is doing it a different way. This is analyzing this signal detection problem and plotting D prime. What this gives us is the time series so that we know that out to about 64 intervening images, the animals do pretty well. They don't control animals. The animals with a TE lesion or a combined lesion have a pretty bad deficit all through all spaces. It made us, well, okay. So the problem we have now is we have an area, TE, that is receives visual information, and it is supposed to be receiving that from TEO. The majority of the information. At least that's what we've been told, led to believe by the unanimous. And we have something that looks more like this, which is some of the response, and this is a guess. We have more, I'll show you some data. The information from V4 goes to TEO and TE. That's the way we've all been brought up to think of this. There's a bucket brigade, you spill your visual information from one area to the next area to the next area. It's been known for a long time that there are projections from V4 to TE, but they always get minimized. People talk about them, but they say, well, but it isn't a very strong projection based on basically no real data. And it's a hard experiment to do, as I'll show you in a minute. But we now gotten into this problem of there's learning going on here, even if TE is remote, TEO is remote. So Sorry. Yes,
So this is really learning related to object recognition versus object classification.
we would call it object discrimination,
This discrimination.
dog versus cat.
Yeah, that's what I thought, too. It may be done computationally very difficult, different to classify the category of the animal versus matching the same.
Yes. Oh, that's true, I can't. I don't have data to talk to that.
But he's not asserting whether TE or TEO is doing either classification or.
No, but in the first task, we didn't behave differently, and in the second task, we got a difference when it was about finding the same. So that's a magic task. So.
Anyway, so we had a couple of monkeys and Mark Eldridge and a young guy who's down Harvest. Pearl did this following experiment. This is an expensive experiment, so we could only do it once each in 2 monkeys. You'll see why in a second. Which is we had 40 training images and 480 test images. The monkeys were passively viewing the training images. And then they went through 1 day of category training with 40 image control images. The day after that, they were given 480 new images. And then they were tested again in passive viewing of the image set. Okay, so you start out with a control of them looking at an image set that they are not actually supposed to be discriminating. They're then given a little bit of discrimination training, a little bit of practice with a new set, and then tested again in a passive viewing situation to see what would happen to the neuron.
There's no reward during passive viewing,
No reward during.
but why would they be?
There is a reward. They get randomly reward for looking at the screen.
Okay,
We do not ask them to fixate. They just, if they're facing forward, they can have a reward irregularly.
So on the second day of passive viewing, it's the same set of images as on the first day?
Yeah. And it's the same set that they got trained on, 40-40. So one slide got left out. Sorry. I left out the result of this, which is that the average response on the post-training test was about 20% larger than the average response pre-training. So it looks like that only occurred in TE. In TEO, there was no learning. And I'll show you more data about that now. Here's a full stimulus set for a learning experiment. Be careful. The cat-dog pairs, which were on the vertical axis, are now on the horizontal axis. And the cat-like and dog-like images are on the vertical axis. Yeah.
So in the passive viewing, did you look at whether responses got sparser on the second day?
We haven't done that.
It's like with Brunel and you guys as a collaborator, have a result like that in order to.
I'm sorry,
They have a paper claiming that.
who knows?
Nicholas Burnell and the monkey physiologist, I'm blanking.
It's something, I mean, we have the data, it's easy enough to check. This is now monitoring the recording in these two areas. This slide should be first. Here we have Utah arrays. Bring everyone up to date. Just think of it as an array, 2x2 array of 100 fine electrodes that you get to put in into the brain, into the cortex once. These are very fragile. You can't recover them once they've been handled.
So a bed of nails.
A bed of nails, and they've been referred to frequently that way. They get pounded in with an air hammer. Because, well, 100 electrodes sitting on the pin, it's significant resistance, and you can't just easily push it in. And all this is demonstrating is that the number of neurons across days is pretty stable. The number of neurons we record. These electrodes, these Utah arrays, do not, in our hands, record the same units in subsequent days. We treat every day as a separate sample. This is what happens. This is the monkey's behavioral performance, which is day one, the correct rate is 55%. And over 9 days, this monkey got up to just under 80%. And this monkey is a little bit smarter at the beginning and gets up in about 3 or 4 days to something like 80% also. That's the behavior. What happens to the neurons? Let me skip this. It's just too difficult. So this is one monkey and the other monkey. This is the purple line is the area TE data. The black line is the TEO data. And in these two monkeys, I have to say, I'm showing you these data and I'm going to claim something I don't trust. In terms of classic statistics, there's no question this, the data in TE for this monkey got better. The number of...
That's a TE lesion.
No,
No,
it's recorded in TE.
So it's all intact, all the.
Yeah, it's all intact. Okay. And this, in conventional statistical terms, is significant. It's not good enough to make me happy. So we're doing this experiment on a couple of more monkeys. It's a very tantalizing experiment, but it's needs some work. These. Okay, so roughly what we tentatively concluded is that TE neurons reflect the behavioral learning the monkeys don't. And the TEO neurons don't. Yes.
So can you say something about the function of TO and TE as a unified group where one has to do something that's learning and the other is not?
Well, so that's why I went through all that fooling around showing you the lesion results, because the lesion results leave us with this conundrum that if we're doing the cat-dog discrimination test, it looks like TE and TEO are working together.
Yes.
And a partial lesion of either of them causes a mild deficit, and a complete lesion of both of them causes a severe deficit. In the recognition task, one, the recognition task, the other one, the one that in fact is, well, let me go there. Then the recognition task, it looks like TEO is not playing any role in this behavior. This raises a question that I keep asking my postdocs for an idea about, and I'll ask you guys about, which is, what is that tissue doing when the animal's doing the recognition test? It doesn't seem to be contributing to the behavior. Do you believe the neurons all went silent?
So,
For the period that the monkey started the recognition test? I doubt it.
so the question was whether it's a serial bucket or a parallel. But if it's recurrent, there may be something. So how much recurrency?
Yeah, it's got something to do with what we're thinking about. Let me show you one, the last bit of data on it, and then I'm going to stop. Which is, well, I'll stop unless you guys want to see something else. But the last the most recent thing we've been doing is trying to look at the connections from V4 to TEO and TE. Now, that could be done using classical anatomical tracing tools. But we've discovered, we've been doing a lot of work with viral constructs to try to silence neurons reversibly, so that we could test hypotheses about what kind of coding you were seeing under different circumstances. But it turns out the virus, one class of virus for us is turned into a gold mine for anatomical studies. And these are viruses that are, yeah, okay. So here's a cartoon of a neuron. This is the axon terminals. And we're going to guess that some are in TE and some are in TEO. And the cell body is in V4. Okay, the hypothesis is there are enough neurons that go from V4 to TE so that you don't need TEO. Now, can I measure that? Not completely. But we can do something about it. You take one of these viruses, and in this case, don't worry about what it is, but you label it with a green, you put a gene in for a green fluorescent protein, GFP, this famous colored. And then you take another set, another construct, and you put in a red one, a gene for a red one. And now you inject the green one. And the third thing is this virus, this Fuji, has a couple of genes from rabies. Now it's not a replicating gene, so there's no danger in handling it. But it turns out it gets recognized by the brain, by cells, as having this rabies genes, and it transports the product from the terminals, the axon terminals, to the cell body. So you can see the site of origin. If you inject this virus at the terminals and wait a few weeks and then collect the tissue, you can see the cell bodies, and I'll show you a few of them. So the experiment we did was to inject the green one here in TEO and the red one in TE. Wait a few weeks. These are the injection sites. Some of you will be able to see this, some won't. But what we're looking at here, this is V4. This is V4. This is actually V2, V3, which I'll make a comment on in a minute. Here's V4. At V2, at the V2 boundary, it just stops. Here it is in another part of V4.
Barry, could you should point those out with your hand, because the.
Yeah.
Sorry.
The laser just so bright.
Yeah.
Or can you use your mouse, the mouse in your Mac so that people on the Zoom can also see you pointing? Yeah, that works.
Yeah. Sure.
You can also go on the left corner and click the laser button.
What?
The mouse is fine.
The mouse is okay.
Mouse is great, yes.
Just don't press the
You
button.
have to stay away from tapping. Here's some V4, V3. There's some V2. Here's V4 with red and green. This is V4 again with red. The red is harder to see, but it's there. And essentially what we see is a mixture, is little bands of green cells, often right next to a little band of red cells. And so what it looks like is there is the strongest projection looks like it from what we've done. And I'll give you a caveat in a minute. You don't know you injected the same amount of virus. That's the downside. Now, we did inject the same amount, and the titers, effective titers have been tested.
Do you see the yellow?
Yes.
Which is both?
Yep. Yeah. There you are. Yellow. Is that what you were asking?
Yeah.
Yeah.
So
Okay.
that's a minor component, it looks like.
Yeah, it's significant. I mean, let me just skip to and then end. So we can do some kind of cell counting. And basically, CEO projecting neurons are the most common class. This is data from 2 monkeys. The TE projecting are pretty easy to find. And the bifurcating is a lot, there are a lot fewer of those neurons, but they're definitely there. And so we've ended up with sense. And so now what we're trying to do is develop some constructs that will allow us to silence. We know how to silence the bifurcating neurons, the yellow ones. That there's a good trick for already. The other issue is, can we silence only the ones going to TE or only the ones going to TEO and do a behavioral experiment?
These are 2 monkeys?
That's 2 monkeys.
So,
And I mean, those data are not bad for 2 monkeys in the anatomical experiment. Actually, I have one other curiosity. Oh, let me just say, the other thing we saw for free was the amount of how strong the projection through the corpus callosum is. That's what this is showing. And you get a lot of cells from the ipsilateral side in the contralateral TE, and a lot, and even more in TEO. The other thing we found were a lot of projections from V2 into TEO. So anyway, let me stop there. Mike.
yes, a couple of questions. So the bifurcating neurons, is their fraction less than chance?
I can't answer because we didn't do that correctly. It's a little bit tricky to know how to do that because you don't know what the denominator is for easily. I mean, you can just use the raw counts and assume you got it right. But there are few enough of those so that a fluctuation of a few of them may give you a different answer.
And also what layer were those neurons in?
Can't tell you. I think 3, 4. Anyway,
Okay, so. Any questions?
And all the CNNs were inspired by this feed-forward architecture and visual cortex, right? When Jan started developing this stuff, I know he had this in mind, even though he denies it. You know, he discounts it greatly right now, like it's, I wonder what we can still learn, right? There's some skip connections in cortex, but now it's used widely in all.
Well, you saw V2, there was some V2 projected directly to, and by the way, I think Kathy Rockland found some.
yeah, no, Leslie Erlander and Kathy Rockland both knew that.
Right,
What they didn't have was as clear an idea of whether it looks like, I want to be careful what I say here, but whether it looks like there are a fair number of neurons. The best I can say is you don't have to hunt for them, they're there. And
well, look about 5% from your percentages.
yeah, I guess.
Okay.
I mean, just one comment. I mean, if you look at the classic neuroanatomy papers like Feldman, Van Essen, and Kennedy, this whole idea of the bucket brigade is just absolutely not true. I mean,
The sequence of going from V1 to V2 to V3. Okay, so let me introduce myself. So I'm based in San Diego, and I already mentioned that I share a faculty position at both the Salk Institute and UCSD, which are across themselves. What you see here is the view outside my office. And as you can see, there's a cliff here. Actually, you can't. It's one of these infinity things. It's a 340-foot cliff, literally around the mesa. And just to get an idea of scale, okay, do you see these two little black poles? Those are humans. And
There's some gliders over there too, most likely.
the what?
Some gliders.
Oh yeah, if you go over to the right here, there's a glider port. So there are people with paragliders and hang gliders that jump off the cliff. And because there's an updraft, they can go back and forth for hours. So this is really, this is a great place to be.
And also, Jack, if you go look down, there's a bunch of naked people.
You don't have to look down. I actually run on the beach. Black's Beach, it's Vegas Beach, it's a new beach. But it's optional. Okay, so I'm going to be telling you two stories.
There you go.
Wow. Okay, so I'm going to be telling you two stories today, which have been developed in parallel in my lab. And something really amazing happened this year. The two of them collided. That is to say, a link between them, which is really exciting because that link turns out to be the transformer that you're all familiar with, large language models. And I gave a talk here on that last year. Okay, so the work is from two graduate students, UC Chen, who just finished up in my lab. She's now up in Seattle, University of Washington, and Dan Keller, who is a graduate student at the University of Amsterdam with Max Welling, who's in machine learning. And he finished up just recently, will be finishing up and going to Harvard for a postdoc. Okay, so being recorded, got it. So we're this is it's a way to put into context what you're going to be hearing. Looking at levels of investigation from molecular all the way to the central nervous system. We're going to be focusing primarily on what's happening up here at the systems level. We're both in experimental data as well as models, but it's going to be informed by what we know about properties of neurons and networks, and we'll try to explain that this might be a way of unifying a lot of these levels, this approach that we're going to be taking. So, the title was Hippocampal Replay. So what is hippocampal replay? Well, it's been known for a long time when a rat moves along, in this case, a one-dimensional kind of a maze. Mazes are with people in the hippocampus business is just an arena. And normally it's 2D, but here's 1D. It's running back and forth. And what you're seeing here is recording from 13 different neurons. As the rat moves across, it's color coded. So the red spikes here occur to the right, and the blue ones earlier, right? Now, here's what is really quite remarkable is that after running back and forth, and these place fields actually, they appear almost on the very first time they run through. So it's they call it one-shot learning. You put the rat in a new environment, you get a different set of place fields for the same neurons, actually, will be in different locations on the new environment. Now, so here's what happens. When the rat, this happens primarily during sleep. When the hippocampus is in a state called sharp wave ripples, and you can see here is a sharp wave ripple lasts about a few hundred milliseconds. It actually replays the whole sequence.
That shows the time scale acceleration, right?
And it's about 5 to 10 times faster than real time. Yeah, so you see here, it's a couple of seconds, here it's a few hundred milliseconds, right? Okay, so interestingly, sometimes it goes in the opposite direction, which is bizarre. So, this is all about learning sequences. Somehow the hippocampus has learned a sequence. And that's what we're going to be focusing on. This is what we want to be able to understand is how does that happen in the hippocampus? Where does it happen in the hippocampus?
But when the rat runs in the other direction, the same place fields light up on the reverse?
No, it's a completely different set of place fields.
Oh, in the other direction?
In the other direction, it's a completely different set of place fields, yeah. It's different from a 2D maze where you see place fields that doesn't matter what direction you're coming from. And that's because you have experience in all the different directions. Okay, so generative models are a big deal right now in AI. And so the hippocampus is a tiny little model of a generative model, right? It's generating sequences. And it may have something to do with navigation, it has something to do with reports, looking around the environment, where are things located. And we're going to try to model it with a recurrent network model. So first, let me give you the anatomy and show you how we're going to model it. So the input comes in to, this is by the way from Cahal. You heard about Cahal in Barry's lecture, very famous neuroanatomist. And what you're seeing here is not a photo, it's actually a drawing. What you do is you look through slides all during the day. These are Golgi stained neurons, but in different slides, he would later at night, he would sit down and draw pictures of what he saw. But it's really, this is art. This isn't just science, it's art. Okay, so here's the input, the dentate gyrus. It projects to another area called CA3. It's a highly recurrent network. Those neurons in turn, these are pyramidal cells, project them to CA1, and CA1 projects out. And the entorhinal cortex is both the input to the hippocampus and the output. So it's a loop. And what we want to do is to, first in a block diagram, is to model different parts and how they interact with each other. So here's the entorhinal cortex. You've heard about these different layers. By the way, the 2-3 layers in entorhinal cortex are very highly recurrent and a lot like the CA3. The dentate gyrus here is getting input from directly from the entorhinal cortex that projects to CA3. This is the trisynaptic arc. The CA3 is recurrent, and we're going to model it with a recurrent network. It projects to CA1, which then projects back to the layer 5, and you've heard about that from Barry. So this is great. This is actually going to connect with all the talks you've heard so far. Now, in addition to this trisynaptic arc, which everybody focuses on, there are other just like Barry was telling you, there are these skip connections, right? So there's a direct connection from 2 to CA3. There's a direct connection from layer 3 to CA1, and this particular projection is going to be the focus of the model. So we want to model CA3 in particular, and then what's going on in CA1. Okay, so a couple of facts about these projections. Dentate gyrus, the very small granule cells, and they are very sparse. So it's a sparsification cell. CA3 is very highly recurrent. And then the primary feedback to enteroinal is going to be through CA1. So that's the setup. And the hypothesis here is that CA3 stores sequences. How does it do that? Okay, well, first of all, here's a very simple model. We're going to have a recurrent network. And as the animal goes through a sequence of locations, we want a different subset of the neurons. So there's the place field for the red. Here's a different subset of neurons that have place fields for the yellow place field, and the green and the blue. So here's the model now. It's a very simple model. It's called an autoencoder. This is one of the earliest neural networks, which was the goal of which was to reproduce on the outputs the input. So you go through, usually it's a layer of hidden units, which are not recurrent. In this case, we're going to have a recurrent network in the middle. And we're also going to change the output. We're going to ask it not to reproduce the input, but to reproduce the next input. So it's predictive. It's got to predict the next, and this is just the loss function that we have. It's got to predict the next input coming in, right? XT comes in, and then it's got to, CA3 predicts the next one coming in.
So CA3 is the reservoir.
It's what?
Reservoir neuron. I mean, reservoir computing.
A reservoir.
Oh, yes, you're going to hear that it's not a reservoir network.
Okay.
It looks like it is. Actually, if you look at if you see it, it's actually hippocampus is really quite remarkable. It's archicortex, unlike the layered cortex. And it's very broadly, the connections between the pyramidal cells and the projections are very broad, very associative. Right. And it looks like it might be a reservoir network that you don't know is a random network, basically, and it's chaotic, right? But you'll see that after training, this network is not chaotic. It's far from chaotic.
And what's this current and predictive loss? That's how you train it, right?
Yeah, okay, current, this is the traditional autoencoder. The input comes in at T-1, and the output, the CA3 is trying to, the recurrent network is trying to predict the same thing it got as the input, right? That's the traditional autoencoder. And there's a set of ways you have to train in order to be able to do that. But you see, the predictive is you get XT, and the weights now have to predict the future input, XT plus 1. XT plus 1. Yeah, so that's what I mean by prediction. And by the way, this is called self-supervised, right? Because you don't have any labeled data. The data is just coming in through the sensory system, and now the data flows through, and it's abundant, tremendous amount of data coming in through the sensory system. Okay, well, so we use a traditional backprop network, backprop through time because it's recurrent, and so we train it up on an environment consisting of a bunch of inputs, which are the place fields, and the neurons within the autoencoder. There's about 200 of them. We line them up according to their position and where the place fields are, and you can see that it is in fact fading away. Well, the battery's just what?
Terry, I've got one.
That's okay. I can point. Okay. Oh, no, they won't see me. Okay. And actually, where do I find the?
Go down in the left corner.
Left corner.
Bottom left.
Yes, from here.
Right.
To the right.
No, in the left corner, but it's the third circle.
The third circle.
Yeah.
Oh, okay.
Looks like
Now I see it.
there's a point out.
And I do the laser. Got it. Thank you. Okay. So now we put it into a new environment and do some more learning. And what you can see is it learns the new place fields. But compared to the ones in the first environment, they look like they're randomly located, but we can reorder them to look exactly like the left. So that's the way that we do the training. Okay, so now that's great because now we have a starting point. And by the way, I'm sure you've all heard that, oh, these neural network models, these deep learning networks, they're black boxes. Well, that's because nobody's looking into them, right?
No, we look into them, but we never understand what they mean.
Well, that's because you're not looking closely enough. No, you have to analyze them. These networks have to be analyzed just like you analyze brain networks, right? I mean, that's what the point is, not just to have get it to do the task. The question is, how to do the task? And that's what we're going to do. That's what we're going to try to do, is to reverse engineer this little network. Okay, so what you see here is two examples of replay and another one for prediction. In the case of the replay, we've trained the network and now we've given a little random input. And as you can see, it produces the whole sequence because it learned to generate that sequence during the backprop phase.
Sorry, hold on, you're predicting ahead in the hippocampus. This is your autoencoder model of the hippocampus.
Yes.
Yeah.
Yeah, it's been trained as an predictive autoencoder, and now you're seeing that it can replay it just like the rat, hippocampus.
Right.
And in fact, if you have several, and we trained it on two different environments here, you can now give it two different inputs, say with two different ends of the maze, and you just give it one input, and it will give you the entire sequence. So you can store multiple sequences into the same network. Now, and here is where magic happens. Let's look at the weights, right? And let's reorder them according to here's the 200 pre and post synaptics. So this is a weight matrix, and the positive weights are in red and the negative weights are in blue. And what you can see is that it's diagonal, right? In fact, if you do projection along the other diagonal, you see there on the bottom for both environments, it's a Mexican hat. And that's a very traditional form of connectivity. And you'll see in a minute that it has very nice computational properties. The other thing I want you to notice is that these matrices are not dense, they're very sparse. And that's why you can store more than one sequence into the same matrix. Now, this is a tiny matrix compared to the hippocampus, the number of neurons in the hippocampus. You have hundreds of thousands. So that means you can store, many sequences in the hippocampus. In the case of the rat, actually humans, our hippocampus is actually not that much bigger than the rats relative to the rest of
Let's
us.
say, Terry, doesn't look that sparse to me. It looks like at least 50% of the neurons are connected to each other.
No, there's more white here. I mean, it's not even close to 50%.
Okay.
But that really depends on how many things you're storing and so forth. Okay, now here is something that we noticed. Now, let's go back to the diagram here. Okay, so the way that we did the training is to once CA3 produces input to CA1, we compare it with the input coming from entorhinal cortex on the next time step. So it's a direct connection. It's a CA3 input now is getting compared to the next time interval. And it's what you're doing is reducing error. It's error reduction, just like we have in many other algorithms, like, for example, in the dopamine cells, it's our error prediction learning. This is error prediction learning. It's exactly the same kind of algorithms that we're doing here with backprop.
Well, hold on, sorry. Could you just say how time is running? So each of the entorhinal cortex layers, you can think of as a vector of activity at time t-1, right? Right. And that provides the input. So that goes into dentate and then CA3. Right. Let's say it's coming in at T.
Let's just say it comes in at T. Right. Let's just say a constant delta T between each of these areas, right?
I see. And then how many recurrent iterations do you do?
No, we just have one delta T because it's got to predict one ahead. It's got to predict one ahead. And that means when CA3 comes out here, it's trying to predict what is now coming in to the entorhinal cortex at T+1. Right?
I see. So entorhinal is sending it's sending T-1 or it's sending T? Because it's getting X of T from CA.
Actually, we actually analyzed some data from real hippocampus, and you're going to see these T's in a couple slides. Okay. But the point is, all you have to know is that you're trying to predict the next input to the hippocampus.
Mm-hmm.
So was White viscosity and shape just by naturally training using background, or was that some L1 regularization of heuristics that encouraged it?
It was just a vanilla background through time.
Okay.
Yeah, in the technical details, we can go into later.
Very quickly, can you say predict the next input that you predict where and when, or just?
No, this is just the hippocampus actually is deeply embedded. By the way, it's an input for all the sensory cortices. It's the highest level, and it is very abstract. By the time it gets up there, it's getting all kinds of inputs, all the sensory system. It's integrating it somehow, and all we're doing here is trying to predict the next input coming into the hippocampus. That's all we're trying to do.
Fixed space, right?
And some of it is information coming from the visual system, some is coming from the auditory
It's
system.
not clock. That's what I want to know.
It's what?
It's like you see what I mean? You predict like the
A clock. No, this is all happening feed forward asynchronously. It's all asynchronous. There is no clock. We call it delta T in our model. Like we have discrete time in our model, but in the hippocampus, it's all asynchronous.
Okay. Thanks.
Maybe this is not a good time to ask this, but I'll ask it and maybe you can tell us.
Okay, go ahead.
What is the historical origin with respect to the rest of the brain development?
It's a very ancient structure. It's archicortex, which means that it's 3-layer. There's 1 layer of cell bodies, and there's superficial neuropil and another neuropil which is an output underneath it. Yeah. You can find it in, let's see, all mammals. Let's see. I guess there was an equivalent in some other lower vertebrates, right?
Yeah, well, it's the limbus. It's where the cortex joins the limbic system. The hippocampus is there right at that edge. So the limbic system, when you're fully in it, has no layers at all.
Right.
And then you have this hippocampus, which has oriented cells with apical dendrites, but doesn't have. It has maybe 3 layers or no layers, and then you have the normal 6-layer cortex.
Yeah. So it's a vestige of that earlier. By the way, in lower vertebrates, there is no neocortex, but you do see these archicortex.
Yeah, I was just going to say, in lower vertebrates, fish, amphibians, reptiles, there's no neocortex. The telencephalon is a structure called the pallium, and its microstructure is archicortex. So it's exactly the same as hippocampus in a human. And it actually turns out that people have found in the lateral pallium of a couple different species of fish, there are place cells.
So its role always has been spatial memory?
No. It turns out that with rats, okay, there were first this discovery was made by John O'Keefe. He got the Nobel Prize for decades later. But he had rats running around. They were recording from the hippocampus, and they saw these plate spheres. But it turns out that if you look at other species, you see, in addition to that, more abstract things. Like in humans, it turns out that it has more to do not with where you are, but where you're looking. But in any case, this is a digression, but it's a very important digression because it's nothing makes any sense in biology except in the light of evolution, right? So we're dealing here with a structure that evolves over different periods of time for different purposes. But we think, and this is really the message is that this is a structure that evolved to memorize sequences of actions. In this case, movement through space, or it could be sequence of thoughts, or because there's along the longitudinal axis, there's a gradient. And as Mike was saying, is that it's part of the limbic system, which has to do with goals. What you want to do, where you want to go. Okay, now here's something that is a prediction, which almost everybody in the field of hippocampology doesn't like. It doesn't like this explanation because it kind of ruins their beautiful conceptual framework that oh, these plate fields are telling us something about, where the rat is navigating, where it is right now. But my view is that the rat knows where it is. It doesn't need the hippocampus to tell where it is. It can see. So what is it there for, right? If it's not to know where it is. Okay, here's an alternative. Well, first of all, if you look at the top row here, on the left is what you saw in the last slide, but the top row, you see that the activity of CA1 over time, as you learn through epochs, going through the sequences of inputs, is fading. And it's fading because prediction error is going down. It's going down because that's the goal of the learning, is to predict the next input. If the next input is exactly predicted, then there's zero error, and it should fade away to zero. Now, here's a paper from the Schnitzer lab, who recorded, normally they record for a day or two, and then they go on to an experiment. Here they recorded on the same set of neurons for 35 days, and CA1 responses for some cells, not all of them, phase away. And not only there, but at a slower time scale in CA3. So there's something happening here. And by the way, in addition to the ones that fade, there are new ones that are being formed, right? It's called drifting representations, and this is seen all over the cortex. It's that the representations are not fixed, except maybe in the visual cortex. But if you go to the olfactory cortex, if you go to the visual cortex and rats, the specificity of the neurons are changing over time, on this time scale of weeks and months. Drifting representations. And so we can explain that. We have an explanation for drifting representations. It's because you're learning sequences, and once you've learned it, there are neurons that were the error signals disappear.
Just to be clear, in your model, the input from CA3 to CA1 is subtracted from the CA1 neuron?
You use this, you use that as the error, the difference, you subtract, and the difference is used to update the weights in backprop.
Okay, but the CA3 input is like inhibitory on the CA1, that's why it's.
Well, in backprop, it's just, we just put it in as a negative, right? A negative weight. But it's actually in the loss function. It's not actually in the actual circuitry. In the actual circuitry, it's a positive, it's an input.
So you're not just training CA3 weights, but also CA1 weights, or there's like.
Well, right here, for backprop through time, we're training all the weights. Okay, so this is an artificial, I'm going to come back to this, okay, because we'd like a biologically plausible learning algorithm. We think we have one. Okay,
but just to make sure I'm clear, so you're saying this decay in the model is due to the prediction getting better.
That's right.
So that means that the weights from CA3 onto CA1 have to be negative.
Okay, there has to be some, there has to be some, okay, I guess, in the biological system, you would have to have a negative input from one or the other, Right. Okay, that's a good point. Okay, so this is interesting. So this is one of our predictions that the interpretation is different in this kind of conceptual framework. Is that when you see a response, it means that it's a prediction error, and what it means is that the animal goes into a location and the prediction is bad, and that's why you get a response. But after a while, you become familiar with the environment, the response goes away because you've predicted it perfectly, right? So there you go. So that's the setup. This is technical. Now you saw that the matrix had this diagonal structure, and also if you do a spectral radius, it turns out to be much larger. In other words, in terms of the eigenvalue spectrum, than if you are just predicting the current input. So making predictions is different from just autoencoding. And now we put it through paces. We have a lot of different tasks that we asked it to do. The first task was just to reproduce place fields, right? That's the bare minimum, right? If it doesn't do that, forget it. It's not a good model for the hippocampus. So here, the input in that was our model is a sequence of inputs corresponding to a rat moving in its environment. And the input has, we just hand coded the inputs, body direction, roll direction, path integrated distance, and distance, all of the sort of metrics that the rat might have access to from cortical inputs. And then we just trained it up, and you can see we get beautiful place fields. So yeah, place fields are what you expect with that kind of input. But now let's put it through paces that maybe the rat doesn't see, but just to see how what kind of capability it might have. So we use as an M, instead of the world, we use M-less digits, right? This is the classic machine learning a set of inputs that it was now it's a toy model, but a toy benchmark, but it used to be the one that you basically everybody tuned up their network, their learning algorithms on. Okay, so here are two inputs, and you can see that the numbers look a little different because they are taken from a lot of people writing numbers. And now you train it up, and what you see is you're getting some kind of an average, right? In other words, you train it up, and it actually does give you the sequence. You give it, in this case here, you give it 0, 1, 2, 3 from one of the people that wrote it, and it will continue 4, 5, 6, 7 with the generated average from all the inputs you got. Now, so again, now that we're talking about this is nice, but we like to analyze the network to see what's going on. So what we did was we put the activity pattern in CA3 through independent component analysis. This is a way of being able to dissect out independent patterns. And what you can see is that color coding each digit, in this case, we're just showing you about 0 versus 2 and 3 versus 4. But you can see that they're very nicely clustered. And they come out, these independent components for each one comes out sequentially over time, as you can see on the bottom. And it continues, by the way, it wraps around. It just keeps going. And this is actually a very important point, is that the network is able not just to memorize the sequence, but it can keep going. Rather, it's not fading away. It's not like one of these reservoir networks where the inputs fade with time. So it can't do this. Reservoir network could not do this. So what kind of well, let's do a couple more. It can not just do a sequence of different digits, but it can take a single digit and rotate it, right? So the same kind of thing that you saw in the last slide here on the upper left. But now when we look with these principal component analysis, what we see is that each digit in the activity space has a subspace corresponding to the digit, separate from all the others, just like we saw with ICA for the MNIST. But what's really neat is that it rotates. Each one rotates in the same direction as the digit rotates. And it will generalize to new digits that it's never seen before. So this is telling us that this is a really powerful way to generate sequences, remember sequences, and be able to generalize. Yes.
So first of all, what are the two rows that you're explaining?
Okay, so what you're showing here are two training inputs. The training input consists of a 0 that is rotated by 360 degrees as a sequence, right? So we're asking it to be able to rotate inputs. We're going to ask it here. So here's the prediction.
So, but is the sequence is each image up at n times delta t?
Yeah.
So first one's at 0, second one's at delta t, third one.
Yeah. Oh, that's right. Each tick mark is a delta t. Yeah. In the simulation, and there are two rows because we trained it on two different digits, one rotating, 0 rotating, and then in the second row, the second B prediction, we gave it just the first, and the output was the entire sequence, rotated sequence. It learned how to take the input and rotate it as a sequence of time.
So can this kind of network generate the secrets in reverse order, which is something clearly the hippocampus does?
Yes. Okay. I'm going to come back to that. That's a wonderful question, and the answer is yes. And how it does it is really a beautiful result. Okay. Yes.
Also, it looks like the generalization. You have the cluster generalization problem where you're creating figures like the 7. It doesn't look like 7 coming down. Looks like some composite structure.
You have to squint, but look at 8 below it.
Yeah, 8 is good, but 7 is not good.
Well, this is what we get. It generalizes. It's not perfect, but come on, be a little generous.
So what happens if after the stop an input comes in that is unexpected? Like, does the PCA trajectory increase? Like, do you see that?
Well, that's what we did here. So the 7 and 8 were completely unexpected. They've never seen it before.
But then the next sequence that comes in is something that it hasn't been trained for. The
It
sequence.
has not, what happens here is based on previous training on 2 digits, it's now trying to go through a sequence which is roughly rotating that input.
But let's say you intentionally provide an input which is different from what it has seen before.
It never saw a 7 or an 8. It was trained on 0 and 1, just 2 images. It was trained to rotate those 2 images, and we gave it a completely new 2 inputs that they had never seen before. And it learned invariance. It learned some kind of rotational invariance. Okay, so it's learning something more than just memorizing. It's memorizing a kind of a rotational operator.
Yeah, no, I think I was saying that if your input was other side rotation, that it knows that it needs to rotate it like this, but now it suddenly received an anti-clockwise rotation.
No. If you wanted to, you could train it on anti-clockwise. We didn't. We trained it on clockwise.
Okay.
But this is not what the hippocampus does. It's possible that hippocampus does invariances, but it's probably done earlier in the visual system.
So, okay, one quick question. The hidden units, these are all in C3?
Yeah,
Okay.
it's all the 200 units in our recurrent network model C3.
And how do you get this angle versus angle matrix out of, like, when you look at your C3?
What you do is you look at the where you are. We can take any given digit, and we can look at what the prediction is from from the PCA, the top 3 PCAs, and we can predict what the angle is. That's all.
So the other thing I'm a little confused about is I thought that the prediction was kind of a negative image that was canceling the input from entorhinal cortex to CA1. So
Okay. So again, for that probably that doesn't matter. But if you're doing the brain, it might be that the input coming in from entorhinal is negative, and the back prop, the CA3 is a positive signal.
Okay, so
You just have to take a difference, and there are a lot of ways you could do that.
so you can just flip the sign in your model or something.
Right. It's the same thing with the reward prediction error. I mean, nobody talks about where is this attracted. There's a couple papers out there, but it could be done in a lot of different ways. Okay, but it's worth to think about that because it might give you hints of what to look for, right? So where is that, right? That might be another prediction. We could come up with a simple way to implement that.
Is the network big enough to just learn a matrix that does this rotation? I mean, you have.
This is a tiny network. This is a toy problem. And what I'm saying is that it should scale up to much larger networks with much more complex inputs and so forth. So yeah, this is a small network, and we've in this case only trained it on 2 different sequences. But we could scale it up. But this is where we are.
So folks in the diffusion discussion group have been kind of trying to think through some of these relationships, right? Because it's an iterative process where you're incrementally denoising something, and it's kind of a predicted sequence process,
Right.
plus 1. So I think that's kind of like conceptually a really similar model where you have something with a lot more neurons that can store many more patterns, many more sequences. You have a whole distribution of natural images and sentences that can describe them. So I think it's possible that there's a connection between these 2 things. Like the thing that's powerful here with an autoencoder, my chief complaint has always been you never know what's important. You're reconstructing the background pixel with the same weight as you're constructing the pixels that are part of the 1, and there's no saliency, there's no importance. With predictive coding, you're getting something that matters. You're penalizing only when you make a prediction about the future that's incorrect, and that's a much more powerful model.
Yeah.
Representation.
Yeah, I was surprised it did this. I have to say, I did not expect it to be able to do this. So place fields, yeah, I think it should be able to do that because it's what happens in the hippocampus. This does not happen in the hippocampus. Right? We're exploring the capabilities right here, and we're just trying to push it and see where it can take us. Okay, so one last example of this is just to show you, you can make the task even harder. This is a case where we have these figurines, and it's a video clip, right, with the number of like 10 frames of each one of these figurines. And in the top row, it's walking, and the middle row, it's the hands are going up, and in the bottom row, and each figurine is different, right? So these are unique sequences, unique videos. And the question is, can you train it up on 9 different sequences and then give it 1 at the beginning and then have it then produce all the rest of the sequence, right? And the answer is amazing. It does it beautifully. In fact, with ICA, it just separates each one of these 9 into each color is a different action. This was even more amazing if you think about it, because video prediction is very difficult. A lot of people have tried doing video prediction and it sucks. You can't go more than 1 frame before it starts blurring out. And it just does a beautiful job. I mean, it really is quite remarkable. Okay, now we wanted to test a prediction, which is that the signals that are coming in to CA1, if we compare the timing between the dentate gyrus and CA1, right? If it's not predicting, then it should be delayed. But if it's predicting, then the dentate gyrus and the CA1 signal should be occurring at the same time to record for both locations, right? That's the prediction. So we got a hold of a data set from the Allen Institute, which was primarily visual cortex. They have neuropixels, but it turns out that they had a bunch through the hippocampus. So we have a data set for look at. And so what we're doing is we're recording a dentate from one location, and then we're doing an analysis for like cross correlation or mutual information with locations in in this case CA1. And so we do all the pairs, right? And look at the relative timing. And I'm going to go through this very quickly. So this is what we expect with and without prediction. So here's the entorhinal cortex. 1 time step later, you get to the dentate gyrus. 1 time step later, you get to CA3, and then 1 step later, you get to CA1. Right. So that would be the normal progression, right? If it was just simply a feed-forward sequence, right? But if there is prediction going on in CA3, then the output should not be 1 step further. In fact, it should be 1 step earlier. And if we compare the dentate gyrus this distribution with CA1 distribution, they should be dead on, right? This is the smoking gun, right? If we can show that the distributions and dentate CA1 overlap, it means that CA3 must be doing a prediction, right? Does everybody agree with that? Is that would you consider that good evidence if that happened?
Terry, you're setting us up.
Okay. So here are the results, and I'm going to focus here on the money shot here, which is DG and CA1. Okay, and it actually surprised us. So cross correlation has 0 delay, and the mutual information also has 0 delay, and the mutual information actually has a peak at 0 delay. So mutual information is probably more important than cross correlation. But we got a really surprise for cross correlation is that it was bimodal, and we don't understand what that means. It means that some were ahead and some were behind, right? So maybe a heterogeneity among CA1 neurons that we just don't understand. But there's evidence that in fact there is heterogeneity.
So these are random single neurons in dentate gyrus and CA1,
right?
So there are a lot of potential pairs.
That's where, yeah.
And this is the average among all those pairs?
Yeah, well, actually, what you showed here is the gray are actually individual pairs.
I see.
And so you're seeing the averages in black and then all the individual ones. In fact, if you look at the top there, you can see that there's a distribution, and that's the bimodal modality that you see here.
Right, you can see it, yeah.
Okay, so this, but what's the other surprising thing is that the time delay, the shift is here in between CA3 and DG is about 2 milliseconds in the right direction. Right, so that's the traditional feed forward. But 2 milliseconds is about the synaptic time delay. So it all fits, and I think that this is really. I mean, it's we didn't know what we would see, and here we have something that is consistent with this new theory. Okay, so I don't have time to go into this except to say that we think that there's going to be a biologically plausible learning rule, several learning algorithms going on hippocampus that can reproduce what I just showed you. And the first one goes back to paper, as you can see, 1987 by Jeff Hinton and Jay McCullough. It's called recirculation. And very briefly, the idea is that you have one hidden layer, both feed forward and feed back. And what you're doing is you're comparing the inputs with what comes back, and you train the weights going up. And now you compare the two time points here in the hidden layer, and you then train the weights going down, right? So you're only training on two different cycles, and it doesn't work. Okay, it falls apart. It's just not a very robust algorithm. But in principle, it's local, right? This is a Hebbian algorithm, so it'd be nice if we can actually get it to work. And the answer is we did. And the way we did it is by having, in addition to the F and G are the weights going up and down across two time steps here. We also have a learning algorithm for the recurrent connections. And that turns out to be, I don't have it here, it turns out that if you do the backprop algorithm, it has two terms. One of them is a local term, and another one is the backprop term. And this algorithm consists of forgetting about the backprop term, just using the first order, not the second order. And it works. This is a very tiny network. This was done by an undergraduate, by the way, a really smart girl, Mia Cameron. And it's on two networks. One is a 4x2x4, right? This is two layers, 4 inputs and 2 hidden layer units. And you can see that here are the actual activities of those 2 units. Each one's like a code. It's basically created a code for each of the inputs that you're giving it. You're giving it 4 target inputs, 4 inputs that is the target, and it replays all 4 perfectly. Same thing here with 848, and it's now 8 inputs, and it predicts. This is like the coding with NCA3 for those 8 inputs. So this is again proof of principle that it might be possible to do everything locally, right? Whether it scales up or down, we don't know yet. Okay, so that was an interlude between that part and now the next part, which I have to go through very quickly. When it's point out something that Michael mentioned very briefly, which is that this sequence here that I pointed out, where you go from a very sparse representation of the input to a recurrent network, and then output, and then comparing it with the next step, is actually recapitulated in the cortex with the layers of the cortex, because inputs from the thalamus, at least in the visual system, go to layer 4 and layer 5. And so it goes from layer 4 to a very highly recurrent network, which is like CA3 in the hippocampus. And from there, it goes down to layer 5. And now it can compare the next input coming from the thalamus with the predicted input. So the very same predictive autoencoder might actually be occurring within the cortex itself. And you can stack these, right? B1, B2, B3. So, okay, this hypothesis, if it works out, is beautiful because it means that the cortex is just not, it's not there just to do simple inputs, single inputs. It's there to deal with sequences of inputs and generating them after you've learned with these recurrent kind of learning algorithms to be able to generate sequences. And maybe this is why we can remember songs and music, right? We have the ability to, and it must, these are very complex temporal sequences that we can remember very well, right? Music. And not just music, but a lot of other things that visual inputs and so forth. Okay, so now predictions. Connectivity matrix is a line attractor. Oh, let's see what happened. Let's like this out. Okay. Okay, it's coming. Okay, so. Okay, this is also from our biologically plausible network. It can also do MNIS, as you can see here, with 6 digits, and also rotating MNIS.
So let me
Yes.
In these networks, unlike the Bayesian generative model type of networks that also will generate hallucinations, in your networks, you don't see hallucinations.
We don't what?
You don't see the network hallucinate your characters or different things.
I think it might be possible if you had interference between them. But with backprop, you don't, it separates them in a very nice way. So it could well be that in the real brain, you don't have perfect backprop.
Kerry said long time ago that lots of the diseases, hallucination come from struggling of time. One of your dogs, right?
Right.
Okay, but
Okay, look, I have the second half. Okay, I'm gonna fit it in the next sentence. Okay, so now here is the architecture of a transformer. So unlike humans where you get one word at a time, in the case of the transformer, one of the very important changes that they made was to encode all of the inputs coming in as a single vector, right? So that means that it was all as that propagated up this feed-forward network on the right. You give your query on the left, but then there's a feed-forward network that's generating one word after the next, right? It's a loop. And but it now can have something called self-attention, where it's a matrix which can gate or give you a sense for the relationship between words, the different parts of the input, the vector, and you can learn those relationships. Okay, so here it is. It can be done with text. So this is the vector embeddings. It can be done with audio. It can be done with, that's the beauty, is that it's given the whole string at the same time, and from that it can make much better predictions than if it was only given one input at a time. Very important, I think, the key to transformers. Okay, now, is it possible that the brain is doing something like transforming sequences of inputs into a vector, a long vector? Okay, wouldn't that be wonderful? So now I'm going to go to this other part of research project that I've been working on for many years, having to do with traveling waves. They've been seen in every part of the cortex at many different frequency bands. By traveling, we mean a pattern of activity that moves across the cortex. And I'm going to give you two examples. This is actually the hippocampus. And this was already done by Thomas Siapis at Caltech, 2008.
You have to go out of that arrow, that mouse pointer, otherwise you can't get activated video unless it's part of the click sequence. Does space work?
Okay, let's. So I go out and say,
turn on the videos with that.
Just hit the space.
How do you get out of it actually?
Just click on laser pointer.
Click on laser pointer again.
You're right. That was a top. Okay, here it is. So you're going to see activity color coded by plus and minus. I hope.
Oh, medium up and down.
Yeah.
Anyway, just right.
Oh, okay. What you would have seen is that the peak red moves across the hippocampus in a nice. It's called the theta wave. Very well known that there's an oscillation in 4 to 8 hertz range. And now that everyone thought it was an oscillation, the whole hippocampus is synchronous. No, it's not. There's this wave going across the hippocampus.
Where's CA1 and CA3 here? Is this just one of these areas?
Oh no, the CA1, CA3 corresponds to slices like this. When you do a hippocampal slice, you cut it like this, and there's the 3 neuron arc goes in a circle around the slice. And I'm sure this is not going to work either. Oh, there it is. Okay, now this is what happens when you go to sleep. There's something called sleep spindles, and sleep spindles correspond to this is an array of electrodes on the cortex of the human who has epilepsy, and the idea is to find the focus. And but what you can see is that during this 10 hertz sleep spindle, which lasts about 2 seconds and it occurs thousands of times during the night, but it has a circular motion. It's so it goes from frontal cortex to the parietal cortex down to temporal cortex. That's where Barry was talking about. It circulates like that. It's just beautiful. We've replicated this in babies. By the way. It is even more interesting in babies because they sleep a lot. Okay, so Ryan Muller, who did that study about analysis, we decided to let's look at the literature. I mean, these traveling waves have they been seen before? And the answer was 100 papers who have people have reported this. It's in the literature, and actually people are aware of these papers. It's not like they're obscure. It's that people ignore them, and they ignore them because they have no idea why they're there. Would you say that's an accurate?
Yeah, I mean,
No.
purposes of argument, yes.
Oh, okay. No, there are a few people like Mike, who think about these things, but most people don't. Okay, and I hate to say, this is my personal experience. For example, I have colleagues at UCSD who work on Hippocampus. That's their business. And every time I'm on the thesis with one of their students, I ask a student, "Well, you've recorded these place cells, but what about the traveling waves? What about traveling?" And here's what they say: "Well, that's not my problem." And so that's the normal thing. People just carry on with the way they always did their experiment. They just ignore it. Okay, so the purpose was not just to collect experiments, but also to come up with possible functions. And one of them, I'm going to show you, there are a bunch of possible functions, but that it may be preserving information over time. And I'm going to cartoon it. So here are 3 inputs on the visual cortex in a straight line in sequence. And here now is the wave field. This is something that's been measured and replicated. So at time 0, we see that we have a point activity that is starts. It's like you throw a pebble in a pond. It just starts from that point and it just spreads out. Now what's happening though is interesting. So if you look at the second time point, it turns out that all the information about where it started and how long ago is contained in this wave, and you can read it out very simply because the center of the circle is where it started, right? So the information is there, and the distance, the radius of the circle, is related to the speed of the traveling wave to the time interval. So this is recoding the information about what happened in a brief time ago into a pattern of activity in space, right? This is a conceptual problem that it took me a long time to grok it. And I've had, I sat down with a lot of people and tried to explain it to them, and it's not easy. Eventually, if you just think about it enough, it becomes, you realize that, okay, it will work. And let me tell you why it's very difficult. Have you ever seen a hologram? So hologram is interference fringes when you shine a laser beam at an object. And it's just a bunch of lines that are mixed up. And if you look at it, you don't see anything. But you shine a laser through it and you see the object. That means all the information about the object was encoded in this interference pattern, right? And what I'm saying is that the same thing is happening here, but not in 2 spatial dimensions, but in a space-time dimension. Okay, so what happens now when another input comes in, you get the same thing, and now here's where they cross, and that's the interference fringes. So and you can keep going on, in terms of all the ways that all the things that are happening at different times.
The red dots are happening, the red inputs are happening at different places.
Yeah, different places at different times, just for the purpose of a cartoon. But it could be very complex video coming in, and with all the interference pattern, you can show mathematically all the information has been preserved, even though it's all mixed up, right?
But there's the actual circuitry of V1 underlying this.
Yes, and in fact, John Reynolds, a colleague of mine, actually recorded from awake monkeys these waves going by. It was showed that it affected the behavior of the animal, what the phase of the wave was when the animal had to see if there was a moving object near threshold. But there are 2 different kinds. This is stimulus evoked, but there's also spontaneous waves, right? So it's more complicated. I'm just giving you the simplest version. Okay, now here's the amazing thing. So I showed you that matrix that you saw that was learned through the background through time. It turns out that's called a Toeplitz matrix. And a Toeplitz matrix has the same value along the diagonals. See, there's there's one along that one, and along this one, there's two. And then if you think about that Mexican hat is just a cross section, basically across.
What's a little bossy?
Across here. Yeah, okay. And so it turns out that that very same connectivity pattern was discovered back in the 1996 as it's called a ring model. And so it turns out that what happens is it's called the bump attractor or a line attractor, because it's you get a bump of activity that's sustaining, and it can move either direction with a little push. It's neutral. It's as if there were instead of a point attractor, there's a line, and no matter where you start, you go down to that attractor and you can be pushed very easily along the line. And going back to Mike, okay, if you have a Toeplitz form, which is the kind that we saw that came out of the learned CA3 model, that means you can push it in either direction. You can go forward or backwards, right? So here's the natural mechanism for doing something that might be difficult to imagine. And by the way, another way of thinking about this is that it's a temporal convolution. Right, that's really what Toeplitz does. And there's a whole beautiful mathematical theory about what's going on here. It's a really beautiful way to do sequences, basically, learn sequences. Now, okay, so Andy Keller noticed something. He said that you don't get traveling waves when you have a fully connected RNN, but if you partially connect it, you get beautiful traveling waves. And if you use this Toeplitz matrix, you get not only beautiful traveling waves, but the traveling waves you get going over, round and round and round for a long time. And so here is an example. Now we call them wave RNNs versus the simple fully connected RNN. And you can see these beautiful waves, and you give it a sequence of inputs, and you get these waves being sent out at different temporal intervals. Nothing in the recurrent network, and here, if you do a Fourier transform, spatial temporal, you get a beautiful line, and the slope is the velocity. Okay, now here is something which I was just absolutely astonished. If this pans out, this could explain a lot of things. Okay, so first of all, let's hide the floating control. Okay, so here's a task. It's called the copy task. So you have inputs into your this wave recurrent network, a sequence of binary inputs along the inputs, and then you train it to basically reproduce that on the output after it ends. And you can put in time delays, like 0, 10, 30 delays. And you can see that after training the wave network, it reproduces the input perfectly for all the time delays, right? But if you use a fully recurrent network, look, even when there's no time delay, it's already smudging out. It gets a few of the first two inputs, but then it smudges out. And by the time you get to 10 delay, it basically has nothing left.
So I don't understand. So you restrict it during training to only learn Toeplitz, or I don't understand what the mechanism. You're still training it.
You start out actually with a Toeplitz matrix, and then you train it to do the task.
You initialize the lasers?
Yes.
And then train from there?
For this particular task, yes. But by the way, you can also do it by just giving it a recurrent network that has like 10% connectivity, and it'll do almost as well.
Okay.
For the different delays, you had to train it separately?
Yes, yeah, for each delay, yeah.
Okay, so Terry, will it reproduce an arbitrary pattern of input at any delay?
Yes. Okay, now this is pretty amazing, but something even more amazing is the learning rate. Okay, so here is in blue, this is the mean square error as a function of training steps. And you can see that blue is really chugging along very slowly. And look at the wave recurrent network. It is like 2 orders of magnitude faster training. 2 orders of magnitude. I mean, it's learning on just a few examples that it's given. And it's also just as fast with the 30 delay and even 80. So we're talking here that we can create a huge, this is something that I've often wondered about is, sensory input comes in and where does it go? Is it going to just decay away or is there going to be some place where it's being stored like a buffer? This is like a buffer. It's a temporal buffer. It just sequences over and over. It circulates with the line of tracking, right? With the tuplets. If you have tuplets.
So, but when you initialize the tuplets, is it arbitrary tuplets? So you just have the constraint that the diagonals are all the same value?
It's more complicated. It's actually what we do is we have the input comes into a perfect tuplets, and then it goes to a recurrent weight network, which then learns the sequence. Right. So you pre-form it before it goes in. Okay. So
you're presuming that's there, and is it the Laplacian tuplets or?
Yes.
Because that's a very specialized matrix.
Yeah, no, just like I showed you for our backdrop through time, came up with the same solution. Okay, well, here's another task, which is learning. Okay, you give it 100 inputs, and then it is there's another input that comes in a queue that queues two of the numbers. The numbers, there's real numbers coming in. And what it has to do is remember those two and then give you the sum at the end. I think most humans couldn't do that, but you'd have to practice. Okay, but exactly the same the type of behavior. Just look at the learning. You can get the regular fully recurrent networks to learn it, but it takes much longer, right? Learning is much faster, but by the time you get to 1,000. Sequence of 1,000 inputs. The recurrent network, fully recurrent, can't learn it at all, and our beautiful, wave network learns it beautifully. And so this is when this really shows us is that you can, in a circulating pattern of activity, keep track of the whole history of the input and maintain that memory over long time periods. In this kind of a network model. It's a specialized form of network, tuplets network, which we see in the brain.
Tuplets was just a recurrent box in the hippocampus, right?
Yeah, that's right. So when I showed you in our hippocampus model, it took this random matrix. You start with random weights, and it created this tuplets matrix with this Mexican hat, which is a temporal way of doing sequences at time, right? For temporal convolution. Okay, I'm not going to show you this except to say that this is only two of dozens of tasks we've thrown at these networks. It does amazingly well in all of them. Okay, and I think what's happening is that it's mimicking what is done by hand in the transformer, because what you really want is a representation that is taking into account all the inputs that have come in at the same time, and mixing the representations of the different inputs coming in into a single population code for the whole time history, that whole interval of time. All the inputs that came in over the interval of time are encoded down in the pattern of activity that then you could use for doing different tasks in the future, right? So that is the end of the story. Thank you.
Can you say specifically, like, how did you make these wave RNNs? Like, they just have like an RNN, but like you constrain it to be sparse.
No, it was the backdrop through time. It made them sparse, but in this case, I don't think they were. We only trained one network for one task, right? So it wasn't.
But the connectivity matrix for the recurrent network is this tuplets matrix.
Yeah.
So that's an n by n matrix on the connection. So it's not sparse at all.
No, it's not sparse. The tuplets is not sparse.
Just initial.
But the hippocampal model did create a sparse tuplets, right? In other words, it's not perfectly tuplets because it's not all the values along the each diagonal are the same. Because there are a bunch of zeros.
But are you saying it's just a vanilla RNN where the weights are initialized to the triplets form? Is that all? And then you do the see on that?
Yeah.
Oh, you're cool. That's really cool.
Yeah, I mean, it's something that anybody could have done. Nobody. The only reason we did it was that we saw it pop out of our hippocampus model, and we said, well, let's start with that. Yeah.
I mean, there's some parallels the work on Boston on dense associated memory.
Now, you remember when Hopkins, exactly what you're talking about, remember when Hopkins was trying to change, or people took Hopkins.
Oh, yes,
Trying to teach you to do a sequence and it always fade away after a couple of steps.
right, right.
Yeah.
But then we had the talk a few days ago.
Why does this fade away?
I don't know. We had a talk a few days ago that we're gonna continue. Some people are interested from a guy from IBS where none of the Boston students.
Okay, no, no,
This doesn't fade away from the total structure, it's just pushing it down in time.
yeah, it just, it's a convolution, it's a convolution. But there's a whole history of people trying to come up with networks that could do sequences, and back in the old Hopfield days, there was there's tractors, and the idea was you put in asymmetric connections that would allow you to go from one track to the next. And but those, I think that those worked for a few sequences of a few inputs, but
Yes, no scale.
but it wasn't scalable.
So again, the diffusion stuff that we've been talking about is literally at that, you break your input and output from pure noise to a completed pattern and do 1,000 time steps where you're iteratively denoising along kind of like this trajectory where taking small steps allows you to average across the many directions a random walk might take you and more successfully complete a pattern guided by some.
You know, I would say that's now even more mysterious to me than what I just showed you.
Yes, yeah.
Because I think we have an understanding of what's going on, whereas I don't have an understanding of why it does it, what you just described. You see these photorealistic pictures, and how does that come out of noise? That's a mystery.
So this couplets form, it means if you gave it an input of like a certain random set of nodes are active, then across time, like it just marches.
It marches right down the sequence of states that you trained on. Yeah. Right. It's exactly. That's all it does. That's
Think of it like a convolution, where as you shift along the diagonal, that's like shifting the impulse response in time for a long.
yeah. And actually, you can write down an integral, right? A temporal convolution. You have a kernel, and you have an input, and you'll get out the sequence from that integral. You'll get out the sequence in time. Sequence of inputs, sequence of time through a temporal convolution. And this is the matrix version of that integral.
Yeah, we have 2 questions there.
Okay,
Okay, so there's this work on Hippocampal. It's a strong network of self-organizing, I forgot, but your current network. And there you had like a reservoir full of neurons with all the ratios of excitatory and inhibitory neurons, and he included all the biological possible learning rules like Henry, etc. And they learned traffic sequences and also to generate them once we learned them.
I see.
And also to deal with branching, because we have sequences where the first part is common and then it's split. Right?
Oh, that's wonderful. Yeah, I was not familiar with that paper, but thanks for pointing it out.
Gilpin Trish,
Yeah, Trish, yeah. Who's Frankfurt.
So actually my real question is it not accepted that neuroscientists can accept these results that come from background time?
Yeah, can I add one thing to her? This is actually the same question on.
Well, can I answer her question first?
No, it's, oh, yeah, that's first.
Okay, so, back in the 80s, and we came with backdrop, Jeff and I, working on the Boltzmann machine, and he went to Romelhart backdrop. And we applied them to simple networks of one layer opinions. We showed that you can explain a lot of the population codes that you see in visual purpose. And the neuroscientists really were, their concern was that it was not biological. And I think that in retrospect, it was because they made a mistake between how you got there, right? There are many ways of getting, here's a network that solves the problem, right? Forget how it got there. Biology will take you there. If it's, there may not be many ways to solve the problem. Biology will take you there. That problem will take you there much faster, more efficiently, but you get to the same place, right? So therefore, what you should focus on is the end product, not on how you got there. And now I think it's more acceptable. But a lot of recurrent neural networks out there that do have solved the interesting problems that you can constrain. The more constraints you put in, like if you have, it's just random units, they're all the same. Instead, you can put in excitatory units and inhibitory units, so you can put in more details about their intrinsic properties. The more details you put in, the closer you're going to match the data from the neuroscience. So in any case, I think that we've entered a new era where neuroscientists are more open to looking at the end product. But now, yeah.
Yeah, I was gonna say exactly along these lines. I mean, everything you show is based, I'm doing a devil advocate, right? But I would have the axioms we put, which is backprop is equivalent to the brain. And if we did the exact same work with spiking neural networks, what would happen?
We did it. Okay, so I have a paper, I have a paper that came out in PNAS. This is like 4 years ago. We showed that you could take any network that was trained in backprop, continuous units, rate coding, and you could convert it into another spiking network, integrated fire units that has the same performance.
But backprop based, I mean.
No, you take the backprop base and you convert it to a spiking network, which means that the performance does not depend on having a rate code. You can do it with spiking units. And there are people, in fact, we have a paper where you can actually do backprop learning, the equivalent of backprop learning with totally local algorithms with spikes.
Mm-hmm.
But the thing that.
But you.
No, here, look, these learning algorithms, they're discovery devices.
Yeah.
I mean, but we discovered this tuplets. We didn't put it in, right? And it just popped out. Do you want to solve this problem? This is the best way of solving the problem. But now what we've done now is take it to the next step, which is that not only to solve that problem, but it can solve many other problems. In other words, this is a kind of a universal, it may be a kind of a motif, and you can imagine this being in the brain, something that's innate. You connect up networks into. In fact, that's one of our predictions. When people do connectomes in hippocampus, they're going to see tuplets, right? That's a very who would have guessed if that turned out to be true? And I think it's a good chance. That's us. I mean, we guessed ahead of time.
So what determines the longest interval that you can do this? Is it just the size of the network?
Well, if you have
this plot of temporal
frequency. I don't know the answer to that. My guess is that if you put noise in, it's going to fade away. We did not put noise in, so
yeah.
I think without noise it just keeps going. But that's because we're using 32 bits. But that's a good question. Is what's the extent? And there are many problems that you can imagine having if you have a stacked set of predictive autoencoders, right? You can extract at every level. What you're doing really is compressing. What the recurrent network does is compresses sequences into a new representation. And if you do a sequence of compressions, you can get to the top where just a relatively small number of units can represent very complex patterns at the very bottom, right? So in principle, if this actually works in the cortex as well as it does in the hippocampus in terms of generating sequences, it can open up a whole new way of thinking about the cortex as a sequence generator rather than a simple feed-forward network that is processing an input one at a time.
I have like 10 questions. I will only ask one. You started with the picture from Hippocampal, right? The nice, complicated dendrites that the neurons have, all of the different neuron types and so on. And now the story that you're telling makes it look like that all you need is RNNs with white neurons.
Okay,
right?
so Mike and I talked about this earlier. So it turns out that if you just look at any of the EM images or the reconstructions, that you saw from Barry of the projections, it looks very random. The axons go here or there, and and it looks like spaghetti. But that doesn't have to be random. In fact, we know there's got to be specificity on what cells are connected. And so it could well be that, in fact, not only is it specificity about the way you connect up between different cells, but even within a recurrent network, how you connect up. And that's why if it turns out to be tuplets, everybody is going to be very surprised because nobody would have guessed it, including myself, without having this simulation using background through time, which is non-biological. Look, it's a tool. You use all kinds of tools for doing analysis, right? Why not use another tool for giving you ideas for how to solve a problem? That's all I'm saying. Okay, well, it's late, but anyway, thanks very much for listening to the talk.
And this is a rare treat because Mike Stryker is a guy who was right there at the beginning of developmental neuroscience. And he still keeps doing stuff that's right at the very bleeding edge. So he has a view all the way, extending over, how many years? Long time, yeah, not as long as this.
Not as long as that.
Yeah, so this is a good catch.
So tomorrow, Friday, Steve Zucker and I will give a talk. Steve will give a talk about some work that he and I have done together with another coworker. And but I'll give a different talk tomorrow about some current things. So I really just want this to be useful to you. So a lot of it will be I'll be happy to answer questions. So I'm not going to talk terribly long. But I want to ask, you guys have heard of functional magnetic resonance imaging, right? fMRI pictures of the human brain. What does it mean in terms of the activity of single neurons? Do you have a clue? That's one question. Another question is, how many people know what a horizontal cell is? Well, some people could. And what about cortical columns? Do you know what a cortical? Okay, so you've heard a lot of the terms, and when I listen to these talks, I think a lot of them get really into the weeds of neuroscience, and I'm not sure. What I wanted to do was talk a little bit about what might make it put things more into context. So there are, these lovely pictures that algorithms, which actually, even though it was a case that he looked in the morning and drew them in the afternoon, an anatomist who looked more than 50 years later, 70 years later, were able to find the very neurons that are in many of these drawings. So it's actually a very real depiction of the shapes of individual cells. The amazing thing about Cajal, though, is all he had was his static pictures. And at this point, there was no electrophysiology. There was very little known about the actual operation of the nervous system, except from clinical neuroscience. And yet he could draw these little, he drew these little arrows saying which way the circuit worked. And almost never are those incorrect. So it's really amazing. So I just first wanted to start out with nomenclature that you guys know. I mean, I think most of you, engineers learn about cellular neuroscience. And so you learn about, cells and channels and ionic concentrations. And, you have currents moving across the capacitance of membrane that give rise to voltages. And you know about how the action potential is conducted and what the resting potential is, and how synaptic release is controlled by calcium that gets into the presynaptic terminal, and how the plasticity is induced by calcium that enters postsynaptic cells at the synapse. You know about two major types of neurons: excitatory neurons that release one kind of transmitter, and inhibitory neurons that release a different kind of transmitter. And that there are these other modulatory transmitters, serotonin, norepinephrine, dopamine, acetylcholine, and so on. But the receptors for the transmitters are what determines the action of the transmitter. And there are two major kinds. There are ionotropic receptors, where the molecule that is the receptor is also the channel that has permeability to some particular kinds of ions. And then there are, so ionotropic receptors are often gated by a neurotransmitter and maybe by something else like memory voltage. But their action is usually very limited in time. And then there are metabotropic receptors, often metabotropic receptors for the same neurotransmitter that has an ionotropic receptor. It will also have a metabotropic receptor. A metabotropic receptors, the receptor that binds the chemical neurotransmitter is not the same molecule as the channel that it opens. And there's a biochemical pathway inside a cell that may last for, you know, milliseconds or seconds or tens of seconds or half an hour or longer when a neurotransmitter binds to a metabotropic receptor. So it separates both in space and in time the action of the neurotransmitter. And so the, you know, major types of excitatory neurons they have different shapes, they have different inputs, they have different connections and outputs. And there are a much more restricted number of types of inhibitory cells in the cortex. And they are named by something that they express, which may or may not be the thing that cell uses to do anything. So most of the inhibitory cells release GABA, an inhibitory neurotransmitter, or glycine, which, and either of these can act on ionotropic receptors, so their action is very fast and very short-lived, or on metabotropic receptors. But the inhibitory neurons are named in many cases by the name of a peptide they express. And in most cases, nobody has a clue what this peptide does. It's just a convenient name for these cells, and you can make antibodies to it so you can distinguish one from the other. So the cells you hear about are parvalbumin cells that express a calcium buffer named parvalbumin. And their shapes, they're classified according to their shapes. They're called basket cells or chandelier cells that wrap around the initial segment of an axon and control whether depolarization of the soma allows an action potential to start from the action. There's somatostatin cells, VIP cells, which stand for vasoactive intestinal peptide, renin, and so on. And, you know, this is just a little cartoon that shows the sort of a picture of the different kinds of cells and their name. And the interesting thing about inhibitory cells is they are born very, very far away from the cortex, and they have to migrate a really long distance to get to where they're going. And they manage that. And that's another story. So there are also glia. There are really three types of glia that some people think they have important neuromodulatory effects. There are astrocytes, and all neurons are separated from blood vessels by the blood vessels in the cortex by an astrocyte. So the nutrients and metabolic support get filtered through astrocytes onto neurons. And astrocytes also extend processes into the synaptic cleft. So one imagines they have an important role, but exactly what that role is. The well-established role is that they buffer ion concentrations. So the extracellular fluid has a characteristic ion concentration, and it's important that even locally that be maintained at a certain level, and astrocytes do that. And astrocytes also, if the brain is injured, they proliferate like mad and somehow help the brain to recover. Oligodendrocytes make the myelin sheaths inside the brain. In the peripheral nervous system, the cells like that are called Schwann cells. And oligodendrocytes make myelin sheaths that change the capacitance of the neuronal membrane dramatically. So the same amount of current injected into an unmyelinated axon will cause a small voltage change, but injected into a myelinated axon will cause a huge voltage change. And that allows rapid long distance communication in the nervous system. And then there are microglia, which are essentially, we have lymphocytes, they're macrophages that circulate in our blood and go into all of our organs. And each tissue in the body has its resident macrophages. And the resident macrophages of the brain are called microglia. And they kill dead cells, they invade after injury. And so I just wanted to get all the terms straight so that you heard them. That, there's extracellular ionic concentration is maintained pretty constantly by cerebrospinal fluid that bathes the brain. And it exchanges only very slowly with the blood plasma. And the neurons maintain intracellular ionic concentrations, which are different from that in the extracellular fluid. And there are pumps in the neurons that are driven with metabolic energy that maintain those concentrations. And it's the maintenance of those concentrations that allows the opening and closing of selectively permeable channels to change the voltage of neurons and do all the things they do. So the other thing I think it's important to understand is essentially all vertebrate brains develop on a similar plan, and going a long way into development, you can't tell whether it's going to be a chicken or a fish or a human being. And in the beginning of the embryo, there's something called the neural plate, which folds closed like this and makes the neural tube eventually. The edges of this that propose to fold are very, very special because they're called the neural crest, and they give rise to a whole bunch of interesting, important cells in the brain. And the neural tube folds, and then it becomes segmented pretty much using the same set of genetic pathways that, say, insects do for their segmentation. And so essentially the topology of the nervous system is just a single long tube. The neural crest cells, however, that were at the top migrate away and become a bunch of different kinds of cells, but the most important ones are probably, they become the autonomic nervous system, the nervous system that's not part of the brain and the spinal cord. They also become the dorsal root ganglions, which provide all the somatosensory input from the periphery. Everything from touch to pain to proprioception goes through the dorsal root ganglion. And a lot is known about this development as these neural crest cells migrate away, they pass through different environments which cause them to be sensitive to different factors in those environments that make them differentiate into different kinds of cells. So, but if we're going to look at the brain and spinal cord, essentially it's a big tube that bends, and the different segments of it have different names. Essentially what becomes the brain stem, the midbrain, and the forebrain, and it bends again. But its topology continues to be just a single tube with ventricles. The tube is the word that neuroscience uses is ventricles, and for the name of this tube, that goes all the way down to the end of the spinal cord. And essentially it looks like this. And what's distinctive about mammals is that the telencephalon, the forebrain, gets very, very large. The thalamus is similar, the diencephalon, the optic, the eyes are an outpatching of this tube. The midbrain is pretty much the same in all the vertebrates, and the brainstem, the hindbrain, the pons, the medulla are very similar in all the vertebrates. So basically, if you're looking at any animal, if you think about it like this, they're pretty much all the same. However, if you look at the brain in a person, it's like this. The forebrain becomes the cerebral cortex, and it just gets huge. It's essentially like, its topology is like if you took two Safeway bags, one on the left and one on the right, and crumple them together and stuff them inside the head. you would have this 2.5 millimeter thick sheet that is folded that fits inside the skull and essentially obscures everything below it when you look at it from the side. So, I've talked about the divisions of the brain and so on. And in this brain, as it develops, there are two basic kinds of sensory pathways. There are what the anatomists refer to as the lemniscal pathways, which are the sort of straight through, very strong driving pathways from the spinal cord. And then there are these more diffuse pathways, which Michael Berry called the matrix pathways. And that matrix had nothing to do with math. It's like when you mix paint, there's the matrix, and then there's the pigment that goes in it. So matrix is just the thing in which it's dissolved.
Scaffold.
So
Scaffold, more like a scaffold.
yeah, or a scaffold. Yeah. So if we look sort of from the bottom up, there's the spinal cord, and the spinal cord is very conveniently divided. The dorsal part, the part, if you're crawling on the ground, the part that's up on your closer to your back, the dorsal part is the sensory part of the spinal cord. And the ventral part, the part near the ground, near your stomach, is the motor part, which contains motor neurons that go out to innervate muscle fibers. And then these cells that were the product of the neural crust become the dorsal root ganglia that have all of the sensory neurons in it. And the sensory neurons in each segment of the spinal cord send out a long axon, or you could call it a dendrite. It's pretty, it has properties of both, that receive input from the skin, from the muscles, from all kinds of sensory input, and sends it into the dorsal horn of the spinal cord. And then there are interneurons in the spinal cord. In the medulla, which is just the first thing as you go up the neuraxis, there are special nuclei of cells. And when there's a cluster of neuronal cell bodies, sometimes it's called a nucleus, sometimes it's called a ganglion. Typically, the ones that are more peripheral are called ganglia, the ones that are more central are called nucleus. And in most cases, the nuclei are compact groups of cells concerned with a similar function and of a similar type. However, the term is abused in some cases by people who will talk about the reticular nucleus of the midbrain, which is a whole bunch of different kinds of cells that are very precisely connected in different circuits with different functions, but they happen to be intermingled with each other. So it's almost a misnomer to call that a nucleus.
Quick question.
Yes.
So in the ganglia normally, is there some specific structure there as well?
What?
In the ganglia, is there like some specific structure?
Well, in some ganglia, we talk about the dorsal root ganglia, which is a cluster of neuronal cell bodies. And that has maybe 30 different kinds of cells, some of which are that have different kinds of sensory responses. Some of them like cold, some of them like touch, some of them like hard pressure, some of them like, let's see, I'm gonna turn up my hearing aids a bit, so I'll be able to hear people better. Yeah, so ganglia really is more like just a clump of neurons that may or may not be the same and have the same function. Nucleus in the brain is usually used for a compact group of cells that have the same function of the same class. So, in medulla, there's the control of breathing, heart rate, blood pressure, specialized circuits. In the cerebellum, which is this huge cortical-like structure, it's a thin sheet that's folded over and over and over. It has lots of interneurons. It has very widespread connectivity, and it's crucially important for motor learning. in all kinds of phenomena. And for classical conditioning, this sort of thing that Pavlov did, ring a bell and then give food powder, and after a while, the dog starts drooling when he hears the bell. And that and phenomena like the vestibulo-ocular reflex that keeps your eyes pointed straight ahead when you move your head back and forth. That is absolutely essential to have clear vision. And that reflex is organized by the cerebellum and a nucleus underneath the cerebellum called the deep cerebellar nuclei. Then you have the pons, which has the motor neurons for eye movements. It has neurons that respond to audition and the vestibular system, face movement and sensation. And the cerebellum is very closely connected with pons. So interestingly, in the pons, there are specific, and this is really recent, but anybody who gets a chance to hear a lecture by Sylvia Arber from Basel, from the Friedrich Nietzsche Institute, you shouldn't miss it, because she has discovered a series of microcircuits in the pons, which control all kinds of different movements, which are essentially the command motor movements to make mice groom, to make mice eat, to make all kinds of different sort of fixed action pattern movements. And there are what?
Scratching my back is in the pons?
Probably, I don't know about your back, but at least in a mouse, that all of these movements that she has measured and done with video are produced by specific circuits that are organized there. So if you stimulate with a stimulating electrode, you don't get anything interesting. But if you specifically infect the neurons in one of these circuits, and we know how to do this for some of those circuits, and then activate them optogenetically or chemogenetically, so that you're just activating this circuit, you can get a mouse to go over and over, because that's the circuit that makes the mouse do this. So it's truly, it really is true that we have layers and layers of motor control. And in the brainstem, there are circuits that really do very interesting, complex movements. I think that work is real triumph. So then going forward, and this is still true, we're talking about all vertebrates still. We have the superior colliculus, which has the command motor neurons for eye movements. So when neurons in one of the layers of superior colliculus fire at a certain rate, that will produce a movement that moves the eyes. Moves the eyes a certain amount in a certain direction very rapidly. So these are called saccadic eye movements, not smooth eye movements, but really rapid ones. So it's very different from what you would get from stimulating the motor neurons for an eye muscle. Because if you stimulate longer or harder, you just get a bigger movement. If you stimulate in the superior colliculus for a long time, you get jump, 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 jump. And where it goes depends on where in the superior colliculus. And it's in the superior colliculus where there's extremely good evidence that there's a winner-take-all network, so that, why doesn't a donkey starve to death between two equally attractive piles of hay? How does it make a decision? Or, if there are two flies, why does a frog snap at one fly and not snap equally between the two flies if they produce equal activation in the superior colliculus, which also gets visual input? And it's because of a winner-take-all network that is embodied in the circuitry of the superior colliculus. There is another structure, an auditory structure called the inferior colliculus, which is very important for auditory localization. And then in the ventral part of the midbrain, there is this reticular formation. And reticular means like net. And that's where there are many different kinds of neurons and circuits that are doing different things. So the old view of this is it was just diffuse. Somehow the reticular formation, was smart in some way. But if you tried to trace neural circuits, you couldn't find them. Now we know that's not true. There are actually many specific neural circuits, but they just happen to be located next to each other and not in separate compact nuclei. And then there are neurons that have particular neuromodulators, like in the ventral tegmental area, there are dopamine neurons. And there is also another level of motor integration of movement. Then there's the hypothalamus, which has many different chemically specific neurons that secrete hormones and that communicate with each other by hormones that are very importantly involved in appetite, in thirst, in aggression and sex. And there's a wonderful body of work, maybe mostly done by David Anderson at Caltech, having to do with the organization of the hypothalamus and how all of these different highly specific circuits in the hypothalamus interact with each other and with the rest of the body. And this is something that's really still in the midst of a revolution. The whole idea of hunger and thirst and how that's regulated. It was thought to be very simple a long time ago, that you could, that you got hungrier and hungrier, and there were neurons that responded, that signaled how much hunger you had. And that was, hungry was supposed to be not unpleasant. And then when you ate, those neurons stopped signaling hunger, and that was supposed to be pleasant. And all of the work on self-stimulation, there's a series of axons that run through the hypothalamus, the amygdala, the hypothalamus, and forward in the brain. And the phenomenon that was discovered by James Olds was self-stimulation. So if you put an electrode there and stimulate along the medial forebrain bundle, what you find is that animals love it. That is, if you give an animal the opportunity to press a bar and get electrical stimulation in its medial forebrain bundle, and then there's another bar on the other side of the cage where it can get water or food, the animal will almost starve to death rather than give up the opportunity to be stimulated in its medial forebrain bundle. So this is
related to this hypothalamus?
What?
Where is this area on this?
It runs from the area, the ventral tegmental area through the hypothalamus on forward to structures that we won't talk about. But it was the traditional model for people thinking about drug addiction. Well, an addictive drug, what it must do is activate the same system that in an animal you can activate with electrical stimulation. But the interaction of that, it turns out, with drives like hunger and thirst and sex and so on, is more complicated and is only partially understood at the present time. But these things are all going on. So finally, we get to the thalamus, which is above the hypothalamus, but the thalamus and the hypothalamus are not the same kinds of cells. And all of the parts of the cortex are related to a specific part of the thalamus. So the cortex, its topology is a big flat sheet on each side. And the thalamus is more convoluted, but you can unwrap the thalamus in a way that you can relate each point in the thalamus to a particular area of the cortex. And And as Michael Berry said, even areas like the frontal cortex, which we don't know what the principle of organization of the frontal cortex is related to specific areas in a particular thalamic nucleus. But like in the visual system, the principal thalamic relay nucleus is called the lateral geniculate nucleus, and each area of the retina goes to a particular area of the lateral geniculate nucleus, goes to a particular area of the visual cortex. And then going farther forward, there's the basal ganglia, and it too is related to particular cortical areas. And Michael Berry's talk gave an excellent explanation of how the basal ganglia are related to it both for motor and sensory feedback. And essentially, when people get Parkinson's disease, and you've probably all seen the movie Awakenings, where a patient, Parkinson's disease eventually becomes an inability to initiate movement because the basal ganglia are not turning off the systems that inhibit movement. And dopamine plays a huge role in the activity of basal ganglia cells, and you can supply a precursor to dopamine that for a little while, at least, before more of the dopaminergic neurons have died, will allow the Parkinson's patient to move. The tragedy is, of course, the underlying disease is a disease that kills off the dopaminergic neurons. So after a while, supplying this precursor to dopamine doesn't help. But that's something that's activated in the basal ganglia. And then in the cerebral cortex, this is where we start to get a difference among the different vertebrates. And it's called paleocortex, like olfactory cortex, which is related to the olfactory receptors in the nose. There's cortex, which is at the edge of the cerebral cortex, the neocortex, called archicortex, which includes the hippocampus and areas that are involved in emotion, like amygdala. And then there's the neocortex, which in the human, it has 4 distinct lobes. You see it very much in mammals, and has input from the thalamus. So almost all of the human cortex is association cortex. There's a little bit of primary visual cortex, a little bit of primary sensory and motor cortex, and the rest of the brain is what people called it because they didn't know what it did. And the evidence for the primary areas was you stimulate electrically in the visual cortex and you see spots or something more complicated. You have damage to the visual cortex and you're blind in the part of the visual field that's represented in that part of cortex. And similarly with sensory cortex, representation of hands and fingers and so on, and auditory cortex. So, but most of it's none of these things. And traditional anatomists, have divided the cortex into many different areas. Barry showed you one set of divisions today, but the main principle is that the cells in the different layers of the cerebral cortex have different inputs and have different outputs that go to different places. And there are several loops that you can define by tracing these inputs and outputs. But also, if you make sections of the brain, the sections, they all have this 6-layer pattern in neocortex, but some of them have huge layer 5 cells, some of them have cells that aren't huge. Some areas have a giant layer 4, like this, and some areas have almost no layer 4. So that allowed the anatomist to divide the cortex in a whole bunch of areas. So when you read neuroscience literature, people will often talk about area 17. Well, it's just happened to be the number that Rodman assigned to the primary visual cortex. So people will say V1, primary visual cortex, area 17, those are just interchangeable. People will talk about, areas, primary somatosensory cortex is areas 3, 1, and 2. I'm 1, 2, and 4, actually. And, primary auditory cortex is area 43, and so on. But this is just some arbitrary naming scheme. And there are 3 different arbitrary naming schemes which are in use, but nothing is to be conveyed by these except this is when old neuroanatomists were slicing up human brains and decided which areas were different from the others. But because the topology of the cortex is of a flat sheet, you can either computationally or physically, if you put just the right amount of formaldehyde on the cortex to make it so it's a little bit stiff, but not very, you can physically unfold it and make a big flat sheet of cortex that's just a couple millimeters thick. Or you can do this with a monkey. And the rhesus monkey organization is very, very much like the human cerebral cortex, with a big primary visual cortex, a whole bunch of visual areas around it, and so on. You can do the same thing with humans, and this is a case where you would map different visual areas after the primary visual area, map the others with a stimulus that allowed you to see responses via functional magnetic resonance imaging or magnetoencephalography, and you can unfold and flatten the human cortex. But the basic cartoon vision of the visual system is all the input comes through V1. If V1 is damaged, you're blind and you have no conscious vision. And the cells in V1 redistribute the input into 2 basic streams. One stream connected with, that seems, and the evidence I think is pretty good, connected with object identification, and another stream connected with object localization and movement. But these streams aren't completely separate. The cells in them connect with one another to some extent. And so if you define the functions of different parts of the visual cortex by, what's missing when there's damage to them, what behaviors are there, or what they respond to electrically, the temporal lobe, one of the lobes of the brain, which is the one that the object identification stream goes, is associated with, contains at its end, neurons that respond very selectively to faces. So these are firing rate is a function of time. When a monkey was shown, this face, or a distorted thing that doesn't look like a face but has all the same pieces, or a cartoon face, or a face even of a person, but not a hand or not other stimuli they tried. So face areas were recognized in the human temporal cortex. And if you look with functional magnetic resonance imaging and present an image of a face, you see sometimes as much a 1 or 2% change in the magnetic resonance signal in a very specific place in the human brain. And for a long time, this was really a mystery. I mean, how do you interpret a signal like that? We don't have a physical principle that lets us connect an fMRI signal of a certain magnitude to a certain degree of activity of the neurons there. So Doris Chow, who's at Berkeley now, but was at a couple other institutions before them, most recently at Caltech, before Berkeley, decided to make what, I don't know how to get rid of these things that are obscuring us.
There's an X in the top left corner, I think, of the.
What is that?
Let's see.
Top left corner of the box.
I'm on a Wikipedia page. My goodness. I want to get to the PowerPoint. There we go. Doris Chow decided, first looked in the macaque monkey cortex to find face areas, because there were a lot of people were very skeptical that neurons did this anyway. But and she generated 96 different stimuli, some of which were faces, some of which were other body images, some of which were fruits and vegetables, some of which were mechanical things, and some of which were random pixels. And then she just localized the face regions in the monkey using magnetic resonance imaging and put her electrode down through these and recorded from a large number of neurons. And this is one picture from her early paper in this, and human faces activated these neurons very well. Almost none of the other stimuli activated the neurons very well, except this one, which was, I forget what it was a picture of, but it kind of looks like a face a little bit. And the neurons in the face area were activated by that. Monkey faces, cartoon faces activated these cells well. And essentially, almost all the neurons she found were highly selective for faces. So it's very clear that there is some process that results in an abstraction from all of the features of visual input that results in an abstraction that allows essentially all of the neurons in certain parts of the brain, certain discrete little areas, to be selected for faces. And it's thought that there may be similar things for places, for recognition of places. But nobody's confirmed this kind of thing in a monkey yet. You do see place selective areas in magnetic resonance imaging. We don't know what the neurons in those areas are doing yet. And there's lots of really interesting work from Doris Chow's lab about how there's actually a hierarchy of face areas, some of which only respond to a side view of the face or to a particular view of the face, and then ones that are farther downstream respond to a face seen in any orientation. Generally not specific people's faces or specific monkeys' faces, but faces. And there's evidence from a study where monkeys were raised together with sheep, and they responded really well to faces too. So obviously some of this has to do with experience. So another technique that's used with functional magnetic resonance imaging is called looking at resting state activity. And essentially, that I think the field has made a horrible mistake, but taken correlated activity between two areas, and when the subject just lying there and told, don't think of anything, there's some fluctuation in the fMRI signal in all of the voxels of the brain. And you can ask which voxels are correlated at rest best with other voxels. And there's a characteristic way that you see in most people, although the significance of it is really pretty marginal. And this is referred to, and you'll see a zillion papers, since I stopped being an editor of Science and rejecting them, you'll see a zillion papers that refer to this as functional connectivity. And functional connectivity in the brain is to real connectivity as military intelligence is to intelligence. The two areas that have the highest functional connectivity are the visual cortex on the two sides, which have absolutely no connection between them because they're looking at different parts of the world. So, but there really is, you know, these have been characterized as a default mode network, you know, and in, you know, there is a recent paper that actually challenges the analytic techniques that have given rise to thousands of papers about functional resting state networks and functional connectivity in relation to a zillion psychological things. And the validity of those analyses has been, I think, very acutely challenged recently, but you'll read a lot about this. So the other lobes of the brain, I'll go very quickly, just 5 more minutes. The parietal lobe, which if there's damage to the parietal lobe, which is in the area of the dorsal stream concerned with localization and movement, patients just, if there's damage there, patients just ignore that part of the world that's represented in that part of the parietal lobe. And they act as if it's not there. The frontal lobe, it clearly is important for planning actions and remembered actions. And frontal lobe patients who have damage to it, you know, if they're in the waiting room and you give them directions to go to the bathroom, you know, go forward, turn left, and then turn right, they can repeat those directions to themselves, but they won't turn the right way. It's sort of planning action to guide movement is clearly something that's going on in the frontal lobe. That's complicated and we don't understand. And there's tasks that do it. So a lot of work, I mean, Barry mentioned the initial work on the localization and function of the brain was, much of it came out of the Franco-Prussian War, where there were clean wounds that went through parts of the brain. And even up to World War II and the Korean War, a lot of what we knew about what different parts of the brain do came about from the analysis of impairments in behavior and how they were associated with the bullet wounds people received. And then, you know, came microelectrode recording and, you know, and many of the different characterized areas of brain. But the last, almost 20 years now, a lot of work has focused on the cortex of the mouse. And the mouse visual cortex has beautiful maps. This shows maps of where the different colors represent different parts of the visual field, different parts of the Azimuth organization of the visual field. So there's a primary visual area, and there are multiple higher cortical areas surrounding the primary area in the mouse. So they have maps, they have higher cortical areas. Not a lot is known about the different functions of the higher cortical areas in the mouse, but it's an area of very active research. But unlike monkeys and cats and presumably humans, where in, say, the primary visual cortex, there are beautiful columns of cells. So if you go from the surface of the brain down to the white matter, all of the cells will share the same stimulus specificity. They will all like, if the first cell likes edges at this orientation, that will be true all the way down the column. If the first cell preserves the left eye over the right eye, that preference will be expressed to a greater or lesser extent by all of the cells in the column. And so sort of picture the columnar organization with each line segment and color representing the orientation and eye preference of the cell. A cat looks like this. If you look at the mouse visual cortex, it looks like it's all mixed up. But as far as we can tell, the same principles of connectivity, that is, what kinds of connections the cell that's selected for this orientation receives, which in the cat and the ferret, it's clearly a collection of input from geniculate cells whose receptive fields lie in a line like that. That same principle appears to be true in the mouse, except things are no longer organized in columns. They're still selective, but they're not organized. So we don't really know how good a model of the mouse is. So people, you've seen several times at the talks of this meeting, this beautiful map of all the different visual areas of the monkey and what they do and what their names are. And it's not clear exactly how that corresponds to the mouse. What is clear is that the plasticity mechanisms that operate during development in the mouse are that operate in development in the cat and the monkey and the ferret, where they've been most studied, are also operate in the mouse. So there is a well-defined critical period of sensitivity to the effect of closing one eye that that creates plasticity mechanisms that it turns out are operative in normal development, but you can reveal them most clearly by this kind of pathology. And there's a period of very heightened sensitivity, and it only lasts for a little while. And the same thing is true in cats and monkeys and in people too, that including the vision, a droopy eyelid for the first 8 weeks or a cataract for the first 8 weeks of development in a human baby has really catastrophic effects on the visual acuity through that eye. And it's, in a couple of years, it becomes largely irreversible. We can look at the synapses that are made onto the neurons in that eye and see dramatic plasticity when we close an eye. And Jianhua Kang at the University of Virginia now showed what's really going on during this plasticity is when the visual cortex is first driven well by the two eyes, it's driven independently from the left eye and the right eye, because the cells, so when you look at a particular cell that's driven through the left eye, it might be like this, and through the right eye, it might be like this. If you look after the critical period of susceptibility to deprivation, then the two eyes always have the same orientation. So what's going on during this period is that simultaneous binocular vision through an activity-dependent process is causing the cells that are responsive to both eyes to be responsive to the same stimulus in both eyes. So we look at the world through one eye or the other eye, it looks the same. And that's at least very likely to be due to the fact that our cells in the visual cortex respond the same way through the left eye or right eye. But what the mouse has given us the ability to do with a great many genetic mutations and a whole bunch of tricks has made us able to dissect what are the signaling pathways and the cellular machinery that is responsible for this period of plasticity and for the recovery. And obviously one hopes to be able to enhance the recovery therapeutically. So finally, I've gone 2 minutes over, but let me mention there are neuromodulators. So we've talked about these neurotransmitters that activate or inhibit cells very precisely. The primary neuromodulators play an important role, which is mostly not understood. The one you've heard about a million times is dopamine, which is necessary or certainly enhances plasticity in the basal ganglia and maybe in other areas of the brain. But there is norepinephrine, which is generated by a small nucleus in the brain called the locus coeruleus. And when you are excited or active, there's norepinephrine. It's involved in arousal and stress, but it isn't universal. If you read things 4 or 5 years ago, they would say it's just like squirting norepinephrine over your whole brain at once. That's not true. The neurons that distribute norepinephrine to the brain go to very specific places. And if you stimulate electrically in the locus coeruleus, you activate them all. But if you look at what's happening in real life, then you see different ones are arousing different areas of the brain at one time. There's dopamine, which is very important for plasticity. I'm sure you've heard a million things about it. There's serotonin, which is the main chemical neurotransmitter signal that is targeted with psychiatric drugs. So there's at least 25, I think there's 32 different serotonin receptors, and it acts in many places on the brain, but drugs for the treatment of depression and mood disorders are, essentially they're drugs that have slightly different effects on different kinds of serotonin receptors. And all of the serotonin cells that secrete serotonin come from a discrete nucleus called the midline raphe nucleus of the brainstem. And it's still kind of a mystery. I mean, more than kind of a mystery, it's still a deep mystery about how serotonin acts.
But what happens when you stimulate those neurons?
What?
What happens to what the humans report when those neurons are stimulated?
It's a mess, basically. It's not very reproducible.
Okay.
So, yeah, I mean, it's arousing, but it doesn't mimic any normal state of a mouse. So, yeah, I mean, it has not been enlightening. And there is only a tiny amount where you do it on individual cells or, I think that's true in a lot of these areas. If you hit it with a hammer, what you see doesn't mimic anything that's part of normal repertoire behavior. And the last one is acetylcholine, which is the same as the neurotransmitter that's used to make your muscles contract from your motor neurons, but also it's extremely important in your sense of alertness and stuff. And if you smoke a cigarette or if you use caffeine, it's affecting the cholinergic neuromodulatory system. So, there's a zillion new tools, mostly useful in mice, slowly being adapted to be able to work to work on monkeys. I mean, it isn't that long ago. Well, it's probably 25 years ago when the group of us who are using genetically modified mice could all meet in a room half the size of this at Cold Spring Harbor. And, we all traded mice and so on. And what's happened is the tools for manipulating different types of cells and the identification of different types of cells in terms of what molecules they express, which has allowed the targeting of ways to activate those cells or ways to inhibit those cells, either rapidly with light or gradually with chemicals, has really made possible an explosion of really interesting work. And so the mouse brain function is being dissected at a very in very high detail at a very precise level, even with dense electron microscopic recordings, where there are 2 projects to reconstruct all of the cells and all of the connections within a cubic millimeter of mouse cortex. So, we are really just at the threshold of understanding how the specificity of connectivity and circuitry actually operates in the brain. And obviously first in the mouse, but much of the technology that is working now well in the mouse is going to be working in monkeys. And when it works in monkeys, we'll really have an excellent idea of what's going on in humans. So I guess if you want to read about neuroscience, there's a really good textbook that Dale Purvis and his colleagues wrote called Neuroscience. And I think it's about to be the 8th edition, but the 6th edition, pretty much everything you want to know is in the 6th edition from about 8 years ago or something. I think it's pretty cheap. So if you want to have a reference work, just to, if you're interested in almost anything about neuroscience, I think that book is really quite wonderful, and it's suitable for a beginning graduate student course or an advanced undergraduate course. And I guess the only other thing I wanted to say is a lot of progress has been made in neuroscience by studying animals that are really good at something. So birdsong, if you're interested in auditory learning and sound production, the work on songbirds has been enormously illuminating. The work on auditory localization has been just transformed by people studying owls who can hear a mouse squeak a long ways away and swoop down from a tree and grab the mouse silently. Echolocation, of course, bats and so on. So finding the right creature to study what you're interested in, because the basic organization of the brain really is the same in all these creatures. And we think that the plasticity mechanisms, the mechanisms that fine-tune things, are also in common. So, so that's why you'll read about a bunch of different creatures, and it's worth following. So anyway, I talked longer than I meant to, but I'm happy to answer questions. In this afternoon and tomorrow, I'll be around. Please ask me if you're confused about something that somebody said, because when you see these pictures that Barry showed in the different areas of the brain, but it's unless you've studied that, it's hard to understand why. So